a lot of information out of mitochondrial bioenergetics without taking the mitochondria out of the cell. And I'm going to discuss general principles of this, and then I'm going to basically have a case study, which I did some years ago, on neuronal dysfunction, showing the variety of approaches and techniques one could take to investigate mitochondrial function or dysfunction in neuronal cells, but to be a general use, I hope. Let's begin. Okay, I guess that Martin has done something rather similar in yesterday's lectures about the advantages and disadvantages of working with isolated mitochondria versus cells. Mitochondria, isolated mitochondria, you can prepare them from animals of any age. The advantage is you're free of the cytosolic factors, and the disadvantage is that you're free of the cytosolic factors if that's what you're interested in. It's much easier to get reagents and substrates and inhibitors onto and into the mitochondria when they're isolated than when they are surrounded by a cytoplasm and an intact plasma membrane. You can control the incubation conditions, the incubation media, whereas with an intact cell, you're working with the physiological medium which the cytosol supplies. And it's relatively easy to do the electrical circuitry and to quantitate volts and amps. The problem is that you get very limited information on the physiological context. Mitochondria can be damaged during isolation. I spent a lot of time working on brain mitochondria, and they're hideously heterogeneous. They can be from neurons, from glias, from cell bodies, from nerve terminals. So those are the advantages and disadvantages. Intact cells are really the inverse. It's important, they're important for investigating physiological relevance. It's more physiological, of course. The interaction between the cell and the mitochondria is present. You avoid isolation. And one of the most powerful reasons that I liked was that it's suitable for fluorescence microscopy. So in other words, you can use a confocal microscope. You can study mitochondrial function together with calcium, membrane potentials, a whole variety of parallel parameters with the different fluorescent probes. But it is more complicated. Very often, and this is a serious problem, it's restricted to neonatal tissue, certainly in the case of neurons. So basically one is culturing neonatal neurons and investigating them before they're fully differentiated, and this is a disadvantage. And of course, it's less easy to get substrates and reagents in because you've got this wretched plasma membrane in the way. And it's difficult to quantify volts. And most of us use the membrane potential techniques qualitatively rather than trying to get exact numbers. An exception is our colleague Akash Gerentser, who has developed refined techniques I started many years ago to be able to quantify the membrane potential in terms of absolute millivolts. The amps are remarkably simple, and any of you who are using a seahorse know this. So the proton current from oxygen uptake is equally valid in an intact cell, with the proviso that you've got to control for non-mitochondrial respiration, and that's relatively easy to do. Now, there is a third halfway stage, and these are permeabilized cells. And these are cells in which one has used specific reagents like saponin, perfringolysin, staph alpha toxin to permeabilize the plasma membrane. And this means you have access to the mitochondria. You have almost the same flexibility you have with isolated mitochondrial experiments without the need to isolate mitochondria. And if you're careful, the cell architecture retains the overall morphology. There are disadvantages. You can damage the outer mitochondrial membrane if you're not careful and lose cytochrome C. You've lost the cytosol, of course, but that's what you wanted to do. And most importantly, the medium must be mitochondria friendly at the moment of permeabilization. And as you know, that means submicromolar calcium particularly. So you have to have a mitochondria friendly medium for the cells, and then add the permeabilization reagent. So those are the three options, and today we're going to be restricted to the intact cell. And this is really what I've already reiterated. The goal was to develop and apply techniques for measuring mitochondrial function in intact cells that make it unnecessary to isolate the mitochondria. And later today, we're going to be talking about measuring amps in mitochondria. And tomorrow, we're going to be talking about measuring volts in intact cells. Amps in intact cells today, amps in intact cell, volts in intact cells tomorrow. So here's the complexity of an intact neuron, with the proton circuit central driving directly sodium, calcium transport, and indirectly via ATP driving a vast plethora of transport processes across the plasma membrane, channels, pumps, receptors, etc. And the ammeter is equally valid. We are measuring the rate of disappearance of oxygen with intact cells, just as we are with mitochondria, after correcting for any non-mitochondrial oxygen uptake, non-mitochondrial oxygenases. So the ammeter, we're going to be continuing talking about the seahorse today. The voltmeter, we're going to be talking about the monitoring of membrane potential by lipophilic cations, and the cation of choice we use is TMRM, and this is going to be the topic of the problem solving later this morning, so I won't go into detail with that at the moment. So this is courtesy of my good friend Albrecht Dürer from the 15th century, and these are the four horsemen of the apocalypse, and one of these is plague, but I did this slide before COVID came along. And the reason I'm presenting it in this way is I want to discuss four types of mitochondrial stress that cells can be exposed to. And in particular, we're going to be dealing with cultured neurons, but it's really applicable to any type of cell that you want to use. And the particular type of neuron that I'm interested in are glutamate secreting neurons. It was really surprising, really, some 40 years ago, that previously every neurotransmitter that had been discovered had been apparently specifically evolved to be just that, a neurotransmitter. Just think of acetylcholine, noradrenaline, serotonin, dopamine, etc., etc. And then the crazy suggestion came along that the most common amino acid, glutamate, was the neurotransmitter at a vast majority of excitatory synapses, that are those that favor transmitter release. And this turned out to be true. And the question arose as to how this could possibly work. So how could one have a ubiquitous amino acid present everywhere? How could it be a specific neurotransmitter? And the answer was precise compartmentation. This is a nerve terminal, electron micrograph of a nerve terminal. It's very small, it's only about a micron across, and this tells you that actually presynaptic mitochondria are very, very small. They're actually quite tiny. The dots that you see are synaptic vesicles which accumulate neurotransmitter, in this case accumulating glutamate. And if we attempt to estimate the concentrations of glutamate in the different compartments, we might find that it was about 10 millimolar in the cytoplasm, maybe 100 millimolar in the synaptic vesicles. It's actually concentrated by a proton pump, ATP-driven proton pump. But outside in the synaptic cleft, which is where it's released onto the postsynaptic cell, and a resting cell, it's less than 1 micromolar, and so that neuron is dormant. And then when the signal comes to release the synaptic vesicles, the concentration in that local synapse rises enormously, triggering the postsynaptic responses, and that is what basically is neurotransmission. Now, how do you keep these gradients? This 10,000-fold gradient across the plasma membrane, the further gradient into the synaptic vesicles. What you do is to use ATP, so it's energy dependent. Uptake into vesicles is directly energy dependent by an ATP-driven proton pump. Reuptake from the synaptic cleft back into the nerve terminal, back into the cell body, is indirectly ATP-driven because it's driven by the sodium gradient, sodium and potassium gradients, which are generated by the sodium-potassium ATPase, which is sustained by ATP coming from the mitochondria. And that's where the relevance of discussing mitochondrial function is in that system. Is anybody having problems with the reception, or is that?
Yes, there's a little bit.
Yeah, let me just pause once. Everybody mute. Everybody muted.
We didn't get any of that.
Yeah, we didn't. I didn't hear anything that Martin just said.
Okay, can Martin, Shona and Martin mute themselves? We'll see how we continue. Martin? Okay, so let's see, sort of, if Shona and Martin wave a hand if something goes wrong. I am in the depths of the country here, that's the problem. Okay, this is a system which works beautifully when we have oxygen supplied to the neurons. When something goes severely wrong, stroke, cardiac arrest, or traumatic brain injury, falling off a motorbike, then things go catastrophically wrong within minutes in the brain. This is the sequence of events that happens when a region of brain is deprived of oxygen. No electron transport, ATP falls, and the sodium pump fails. The sodium gradient decays, and the glutamate transporter, which is driven by the sodium gradient to take up glutamate from the extracellular space into the cytosol, stops and reverses. Within a few minutes, 4 to 5 minutes, the extracellular glutamate rises 100-fold, from 1 to at least 100 micromolar. And in the ischemic core, the actual volume which is deprived of oxygen, those neurons die really quite rapidly. What is worse is that the glutamate which is released diffuses out into the surrounding cells, the so-called penumbra, and cause a pathological activation of a very important class of glutamate receptors, which are called NMDA receptors, for the synthetic agonist N-methyl D-aspartate. NMDA receptors have a very important role. They are responsible for learning and memory. If you remember anything from this lecture, then it's because your NMDA receptors have strengthened specific synapses which you can later recall, basically, to get the memory back. They have properties which are important for learning and memory. In particular, they let both calcium and sodium into the cells, and they tend to remain active for as long as glutamate is present. The problem is that this is catastrophic in this condition of stroke, because it means the NMDA receptors remain pathologically activated and sodium and calcium flood into the neurons. Now, whether that neuron lives or dies depends on its mitochondria. Can the mitochondria supply sufficient ATP specifically for the activated sodium pump? Can they survive a massive calcium accumulation, massive uptake of calcium into the mitochondria from the elevated calcium in the cytosol? Can they survive an accompanying oxidative stress? So these are all life and death decisions that an individual neuron is faced with when exposed to this pathological glutamate. And the process of cell death is called excitotoxicity. Glutamate is an excitatory amino acid. It increases transmitter release, it's positive. And so excitotoxicity is the problems from too much glutamate in the wrong place at the wrong time. A normal synaptic transmission, the glutamate is elevated for a matter of milliseconds before it's reuptaken, whereas the glutamate can persist in this pathological condition. So we're going to go through each of the four horsemen, because each has relevance to mitochondrial function and dysfunction, and it introduces a whole range of techniques applicable to any sort of cellular bioenergetic investigation. And our first horseman is calcium overload. There he is. Now this is a very early experiment here. I really must update this. This is imaging cytosolic free calcium concentration with the cytosolic calcium indicator FURA2. And we are looking at individual cells here, and we're looking at the response to glutamate. So we've added glutamate, trying to mimic what happens to a cell exposed to pathological glutamate in stroke. And we see two classes of behavior. We see some cells that show a spike, partial inactivation, and then a steady plateau for the length of the experiment. This is a surviving cell. If we switch off the NMDA receptor with MK801, the cell survives. Other cells are less fortunate. They maintain calcium, lowish cytosolic calcium for a period of time, but then the calcium increases through the roof. This is irreversible. It's not brought back by MK801, by the inhibitor. And this is called delayed calcium deregulation. And this is one of the phenomena we're going to look at, the role of mitochondria in delayed calcium deregulation. If we look at an isolated mitochondrion, there's a very familiar process which people have investigated, and some people have devoted their entire careers to investigating, which is that the capacity of mitochondria to accumulate calcium is quite staggering, but it's not infinite. And if one adds an excess of calcium, after what we're doing here is we're measuring the external calcium. This is calcium that isn't taken up. So we add calcium to the medium, and the calcium increases, and then it's taken up into the mitochondria. But after a period of time, the mitochondria give up the ghost, collapse their mitochondrial membrane potential, and release the calcium. And this is known as the permeability transition. And this is one of the eternal discussions in bioenergetics meeting as to what causes it. And I do not intend to go into that complexity at the moment. But the question is, does the phenomenon we're looking at, delayed calcium deregulation, involve the permeability transition? And the answer is almost certainly it does. Let's see what happens in the permeability transition. Here is our nice friendly blue proton circuit, and it's sitting in the inner mitochondrial membrane, And here is the outer mitochondrial membrane of a neuron. And I'm focusing on two components, the NMDA receptor itself and the calcium ATPase in the plasma membrane. And when we activate the NMDA receptor, calcium enters through the receptor. It's permeable to both calcium and sodium. The calcium ATPase works as hard as it can to pump that calcium out again, but it's really relatively weak. It's not very active, the calcium ATPase. The calcium rises, and when the calcium rises over half micromolar, there's net uptake into the mitochondrion. And that process can continue, excuse me, until the calcium accumulation is sufficient to activate the permeability transition pore. And then the proton circuit is short-circuited. The calcium comes out through that permeability transition pore. ATP synthase reverses, that is a dead cell. Permeability transition is terminal. Now, we can see the role of the NMDA receptor in some nice experiments which were done some years ago when it was possible to clone the NMDA receptor. Its structure was known, and one could cut down its activity with siRNA and look at, this is still the cytosolic calcium here, and look at the response, the NMDA response to control cells and to cells in which the calcium uniporter, the mitochondrial calcium uniporter itself, is knocked down by siRNA. So this is full activity of the calcium uniporter. This is inhibited activity of the calcium uniporter. And the calcium elevation in the individual cells or in the mean cells is dramatically decreased. And the extent of cell death in response to NMDA, different concentrations of NMDA, is also reduced. So that's its indication that the calcium uniporter and calcium uptake into mitochondria is playing a significant role in this glutamate-induced excitotoxicity. So we've dealt with the first horseman, which is calcium. Calcium is bad, and the mitochondria very easy overfill with calcium, and you get the permeability transition. It's a bit, let's go back to the previous slide. It's a bit unfortunate that the tendency to measure absolute levels of calcium with calcium-45 isotopes is really passé, and now everybody simply measures free calcium with calcium indicators, and you can load these into the mitochondrial matrix, and you get very nice results. But what you are measuring is free matrix calcium. And one of the extraordinary things is when you get the permeability transition, the mitochondria have accumulated roughly 1 molar total calcium, virtually entirely as an osmotically inactive calcium phosphate complex. So at the onset of the permeability transition, you have 1 molar total calcium inside the matrix as a calcium phosphate complex, and the free calcium is 1 or 2 micromolar. So when you see experiments using calcium indicators measuring matrix calcium, bear in mind they're measuring a minute fraction of the total amount of calcium which can be accumulated by those mitochondria. Let's go on to the second horseman. Here he is, ATP demand. Now, this is a curious piece of apparatus, and I'm showing it here simply because I use this to do the experiments. This is before the days of seahorse. If you can imagine, there was a time before you had seahorses to measure cell respiration. Before that time, it wasn't possible to measure cell respiration. So my colleague Mikko Jacobsons and we devised a flow-through system, a very thin flow-through system where the neurons were cultured on one cover slip, and we measured the drop in oxygen as the medium was slowly transfused, superfused over the cells. and the drop in oxygen gave us the rate of oxygen uptake. And using this primitive apparatus, say in pre-seahorse days, we could do fun experiments. We could, for example, overcome respiratory control in these cultured neurons by adding FCCP, and the respiration increased about threefold. The advantage of a perfusion system over the seahorse is you can perfuse it away again, and the respiration falls, and you can repeat this again, and the respiration falls. Now, instead of adding FCCP, we add glutamate, and we see that the response we get to glutamate is essentially the same as the maximum response we get to FCCP. We can do it either this way around, glutamate followed by FCCP, or FCCP followed by glutamate. And essentially, it's very easy to get to a situation where glutamate is in some way stressing the mitochondria to utilize its entire electron transport chain capacity. So let's think what is happening here. This is our mitochondria, our proton circuit, our plasma membrane. I've put a few more things into the plasma membrane, and the most important thing is I've put in a sodium pump, the sodium potassium ATPase. And while electrophysiologists tell us that the NMDA receptor has typically a 10-fold selectivity for calcium over sodium, in the intact cell, it's exposed to the cell is exposed to 100 times as much sodium, 100 millimolar, than the 1 millimolar calcium. We've already dealt with the problems that calcium gives. So sodium is really flooding in through the NMDA receptor. And the sodium pump, just like the calcium pump, is trying to expel the iron. The sodium pump is extremely active. And in fact, it can utilize virtually the entire respiratory electron chain or ATP generating capacity of the mitochondria. So the sodium pump hydrolyzes ATP to ADP. Here's the adenine nucleotide translocator in the inner mitochondrial membrane. And the mitochondria respire, as we've just shown in that pre-seahorse experiment, by increasing its respiration as far as possible. This was really the first time that it was possible to do such an experiment. Previously, all the focus was on calcium. So there's now a focus on sodium. And we put forward a hypothesis that the survival of an individual neuron depends on it having enough spare respiratory capacity to deal with the maximum energy demand it's ever going to face. And we call this a 100-year flood. It was back in the days of Hurricane Katrina. So how do we test this hypothesis? If survival is dependent on maximal respiratory capacity, we could very subtly and very slightly restrict that maximal capacity with low nanomolar concentrations of the complex 1 inhibitor rotenone. And we see as we do this, toxicity increases dramatically. And this is a relatively minor decrease in electron transport chain maximal capacity. So that fits the criteria that a slight drop in spare respiratory capacity can greatly exacerbate excitotoxicity. And so we're dealing with calcium, we're dealing with ATP. Now, I want to go on to the third, and this is actually Martin's speciality, reactive oxygen species, but I want to just show you one interesting approach one can take, which is something you can do if you're looking at any agent which is stressing your cell population. There's the third horseman. The major cytosolic defense against thiol oxidation, if you like, thiol oxidative stress, is glutathione. And glutathione is of critical importance for maintaining sulfhydryl groups in the cytosol and facing the cytosol in the reduced form rather than being oxidized to disulfide. And there's an interesting compound called monochlorobimane. And monochlorobimane is taken up into the cytosol, where it complexes with and removes glutathione. So it's a way of depleting a cytosol of glutathione. It has one other very nice property in that the monochlorobimane glutathione complex is fluorescent. And you can see the appearance of fluorescence here as the monochlorobimane chelates or complexes the glutathione. And you can follow the time course of increase in fluorescence, which is the time course of decrease in glutathione concentration. So we can progressively and visually deplete a cell of glutathione, and let's see what actually happens to that cell. Again, we are using this pre-seahorse technique, this superfusion technique. Martin suggests I should repeat these experiments at some time in the 21st century with the seahorse, but I haven't got around to doing that yet. But this is basically the classic cell respiratory control experiment that one does with isolated mitochondria in intact cells. You start off, we add oligomycin. Oligomycin blocks the ATP synthase, and respiration, instead of being controlled by proton reentry through the ATP synthase and the proton leak, becomes controlled by respiration just through the proton leak. And then we can work out the maximal respiratory capacity by adding FCCP, and we show a big increase in respiration. So we've got a nice spare respiratory capacity. When we started doing this, we had an interest in Parkinson's disease and the possibility that oxidative stress that was damaging complex 1 was inhibiting electron chain capacity and would therefore show up as a decrease in spare respiratory capacity. What we actually found was very interesting and different. As soon as we added monochlorobimane, cell respiration began to decline. If we waited an hour or so and then added oligomycin, we saw just a very small decrease in respiration. This tells us that the ATP turnover in the presence of monochlorobimane is a small fraction of that in the control. To our surprise, there's no effect on proton leak, and even more surprising, maximal respiration was essentially unchanged. So what does this mean? It can mean either that a monochlorobimane treated cell needs less ATP, or it could mean that the cell needs the same amount of ATP, but the mitochondrion has a problem producing or exporting that ATP. So let's look at the alternatives. One of the ways to further stress the cell is to increase its ATP demand. And you do this, there are a variety of neurotoxins which activate sodium cycling at the plasma membrane. Basically, they lock open sodium channels, which normally spontaneously inactivate after 1 or 2 milliseconds. And now we introduce something rather similar to that, which I've just shown with glutamate, where sodium is flooding in, this time through the sodium channel, and the sodium pump is trying to pump it out desperately. And this increases ATP demand. So we start with the control cell, we add brevetoxin, and we see a satisfactory increase in ATP turnover, basically. And then we add FCCP, and the cell seems to have, up to that stage, survived this increased ATP demand. Now we do the same thing on a cell which is declining with monochlorobimane as the glutathione is used up. We add brevetoxin, we get a rather insignificant increase and a great decrease in maximal respiratory capacity. In fact, it's even less than it was in the absence there. And when we look at the percent of cells undergoing delayed calcium deregulation, it's dramatically increased by that monochlorobimane. So the question, what is it doing? Is monochlorobimane, for example, affecting the ATP synthase, or is it in fact affecting export, the adenylucleotide translocator? And the evidence that it was the translocator came from some very interesting experiments which reflect on something you'll be doing later today when you are measuring membrane potentials in intact cells. We introduced something we called the oligomycin null point test. So what does that mean? If you have, we can think of this with, first of all, with isolated mitochondria. If the mitochondria are generating ATP and you add oligomycin, the proton motive force, or the membrane potential, rises slightly because you have cut off one of the pathways of proton reentry, and respiration slows, and you get less of a voltage drop across the electron transport chain complexes. So you get a very slight hyperpolarization with oligomycin. And if you see that, we are dealing with this later today, or in fact tomorrow, when we're dealing with membrane potentials, that we're using conditions where a decrease in fluorescence means an increase in mitochondrial membrane potential. We just have to accept that at the moment. And in the control condition, virtually all the cells, or half the cells, have a detectable hyperpolarization with oligomycin, which means that they were generating ATP before you added the oligomycin. A number of cells, the change is too small to actually detect. And then we have a third population, very few in the control, where oligomycin does the opposite. Oligomycin depolarizes. How can oligomycin depolarize? If you have a sick mitochondrion such that it is partially depolarized, and it's in an intact cell where you have active glycolysis, the ATP synthase reverses. Instead of taking up protons and generating ATP, it hydrolyzes ATP ultimately from glycolysis in an attempt to keep the membrane potential up. So the ATP synthase reverses in sick mitochondria. And when you add oligomycin, under control conditions, you find there are a very few, perhaps 2 or 3% of the cells in your field are actually sufficiently damaged that the mitochondria are no longer generating ATP, but they're hydrolyzing it. Excuse me. In the presence of monochlorobimane, virtually all the cells show neither an increase or a decrease with oligomycin. It's undetectable. And this strongly suggests that what we've done is to inhibit the adenylucleotide translocator, so we can no longer export ATP, and we can no longer import ATP in order to keep the membrane potential up. So these are healthy cells. These are damaged cells, if you like. And even without this monochlorobimane, simply doing the oligomycin null point test on a population of, say, 100 cells, you can go through the 100 cells and say, so many cells are healthy and generating, mitochondria are generating ATP, and so many cells are damaged and the mitochondria are partially depolarized. More importantly, they're hydrolyzing ATP. So you don't have to measure an absolute membrane potential. This is qualitative, but it tells you whether the membrane potential, the proton motive force, is sufficient for ATP synthesis or is insufficient due to damage. So the oligomycin null point test, which we'll actually model later in this course, is extremely powerful in this condition. Now, we're getting on to the final horseman, and this is age. And as you note from the title of our institute, we are an institute for research on aging. And the question is, what effect might aging have on this mitochondrial function, mitochondrial dysfunction? The regrettable fact, especially for me, is that the tendency for stroke, for for stroke or for heart attack, whatever, increases exponentially with age. And the question is there any sort of reason why the aging neuron has a greater difficulty in maintaining its function in response to excitotoxic glutamate? And the way I did this, and I did this some years ago, was to take a random fluctuating trace. And this trace represents notionally the ATP demand on an individual neuron. The problem we've got is that we're trying to do experiments, acute experiments. We're trying to do experiments that are over by 5 or 6 o'clock so we can go home. And so we're trying to look at the fragility of neurons over a time scale of a few hours. Whereas what is happening during the aging process is a process that's working over a period of 20, 30, 40 years. So how on earth can these acute experiments have validity for this chronic long-term effect? And this random chart, it was the share price of Starbucks some years ago, totally random, represents one of the billions of neurons in your brain, the ATP demand due to neuronal activities. There's a period of fairly high neuronal activity here. And what I've done is, first of all, to estimate the maximal ATP supply that you students, most of you students anyway, have at this sort of age in the 20s and 30s. You have such an excess of spare respiratory capacity that the chances that you'll ever run into an energetic crisis is negligible. As you age, this will very soon be me, not quite yet, for whatever reason, maximal respiratory capacity, spare respiratory capacity decreases, and you can get to the stage just in an individual neuron, stochastically, that you get an ATP crisis. And an ATP crisis for all the previous 3 horsemen in terms of calcium, ATP oxidative stress is going to be catastrophic for that neuron. And that neuron is almost certainly going to die. So it's a stochastic event. One neuron dies, maybe another neuron dies a few days later. It's not a concerted event. And so we put forward the hypothesis that spare respiratory capacity was really important for the survival of neurons in a variety of neurons in terms of excitotoxicity, but also any sort of cell in an energetic stress. So let's take all of this together and put it and assemble it into a flow chart. NMDA receptor activation. We've shown that calcium enters the cell through the NMDA receptor. It is perfectly clear. And we can show that the mitochondria take up that calcium. In parallel, sodium enters the cell. That activates the sodium-potassium ATPase, as we've seen. Mitochondria don't like accumulating calcium, and I haven't talked about this, but if one looks with a confocal microscope at individual mitochondria in a neuron, particularly in axons, you find that the mitochondria swell and they stop moving. They stop trafficking up and down the axon, which is critical for their function. So both the calcium uptake and the sodium pump activation increases mitochondrial energy demand. And as we've seen stochastically, that can lead to an ATP crisis. The calcium uptake can induce the permeability transition. The permeability transition can exacerbate the ATP crisis. Oxidative stress, as I showed you with inhibition of the adenucleotide translocator, can both potentiate the ATP crisis and actually also potentiate the permeability transition. All of this catastrophic cascade ends up as delayed calcium deregulation. So what I've tried to do in the last 45 minutes is to take a mitochondriologist's approach to a specific function, which is excitotoxicity in neurons, and to show the variety of approaches that we use with our bioenergetic knowledge in order to dissect out what is happening, and to look at calcium, to look at sodium, to look at oxidative stress, and then to assemble these all into a single package. And I will stop there. Thank you. And I will stop sharing.
Hi, David. Can I ask a question?
Yeah, hi Pankaj.
Yeah. Could you just sort of repeat the message again of the difference between the maximal ATP supply and the spare respiratory capacity, and which experiment shows really the, how would you test that if you had that question in your setup?
Right. So the question is, how do you detect and quantify spare respiratory capacity? Is that right?
Right, and you're saying that's what's critical for cell survival. That's very interesting.
That's been our hypothesis for the last sort of 15 years ago, ever since we started measuring the energy demand in this particular system, because people hadn't been able to do it previously. And the fact that the spare respiratory capacity is what you've got in reserve for a crisis. And I used to think about, back in Hurricane Katrina days, I used to think about New Orleans and the levees in New Orleans, that
right.
Once they overflow, there is a catastrophe. And the same way, once the maximal respiratory, the spare respiratory capacity is used up, then there's a catastrophe. And we could show that very slightly decreasing that spare respiratory capacity greatly increased excitotoxicity. There's very low Rosalind concentrations, for example. And I don't think this is just limited to this particular case of glutamate excitotoxicity. I think it's much more general, that a whole variety of disorders associated with mitochondria are associated with mitochondrial dysfunction. Martin and I wrote a review almost 10 years ago on mitochondrial dysfunction in cells, and we actually defined, it took about 30 pages to define what we meant by dysfunction. And a central feature is that mitochondria aren't producing enough ATP, basically. So ideally, you want to measure ATP generating capacity with intact cells, because you can't directly access the ATP. You tend to measure respiratory capacity and assume that the ATP generating capacity matches that for respiration. So anything which gives mitochondrial dysfunction, anything to do with sirtuins, for example, modulating cell survivability, one should look very carefully at whether there's an effect on spare respiratory capacity, whether you can actually localize it to a change in that particular parameter, which you can actually measure very precisely. That's in the review which Martin and I gave you all to read. Then we go into great detail about how to measure spare respiratory capacity.
So one corollary of this would be that if let's say you have a disease or let's say Parkinson's, and if there was a way to reduce the ATP usage, right, or somehow the basal metabolic rate somehow, because the spare capacity would be what you can produce and what you're currently using, right? So I was just thinking, what do we know about interventions that lower that basal metabolic rate, and whether they are protective?
What you have to do is to decrease ATP demand.
Demand, yeah, sorry, demand is the right word. Yeah.
So if you can, I don't know of any ways to do it. If you could stably decrease ATP demand, then you'd have a more spare respiratory capacity. But if the crisis is an increase in is then subsequently increase in ATP demand, then yeah, the question is whether it's helpful. But I don't know of any sort of chronic way of decreasing ATP demand in a cell, basically, that would actually work. Any ideas, Martin?
So you can certainly inhibit particular processes that are using ATP, but of course they're using ATP because they need it.
Yes.
So stopping them from working is not really a way to cure a problem. It just gives you another problem. So you could stop cells making protein or making DNA or pumping some iron across the membrane, and that would decrease the ATP demand, but it wouldn't do you any good.
Well, you know, I'm actually thinking of some work John Speakman had in calorically restricted mice. So he shows that that they actually end up, the oxygen consumption is reduced. So I'm thinking of states where you would, you're right. I mean, you don't want to do it, you have to do it in a manner which should be homeostatic to the organism. So it looks like in caloric restriction, the oxygen consumption is reduced. And my first thought was maybe the ATP demand is lower. And because you have to match the food intake with the demand, it makes sense that you reset the organism to a lower ATP demand situation. And that hasn't been talked about enough in terms of dietary restriction, because this idea that metabolic rate lowering is beneficial has been sort of, I think, thrown out of the window a little too prematurely, I think. But I was wondering, what do you think of that? I mean, and the thyroid hormone is one of the drivers, I guess, of metabolic rate, right? So in that context, do you think, would that make sense?
Martin?
Yeah, the trick is essentially the same answer. The trick is to reduce the ATP demand without giving up some function of that ATP. So there may be some processes like futile cycling and glycolysis that have little benefit and use up a substantial amount of ATP. So
right.
Maybe there are some reactions like that you could decrease in principle, and that would be a good thing.
Yeah. The most effective thing is not futile cycling, but actual cycling, in other words, exercise. And
That's exactly what I was going to say.
Yeah, the obvious way to increase spare respiratory capacity is to exercise. You get more mitochondria, you get more. There's a fascinating study that was done many years ago on, I think it was rhesus monkeys. They had two sets of monkeys, and one were couch potato monkeys, and the other were marathon running monkeys. So they had non-exercising monkeys and highly trained monkeys. And monkeys are very, very expensive, and you don't want to kill them, but they gave them a very low dose of MPTP, which ends up inhibiting complex 1 and is used, or was used, as a model for Parkinson's disease. And really interesting that for exactly the same dose of MPTP, the exercise fit monkeys did much better than the couch potato monkeys. Mm-hmm. So, you know, exercise increases spare respiratory capacity. I mean, I'm not very good at this myself in terms of going out and cycling and so on, but it is the single simplest thing you can actually do.
Does it not only increase the number of mitochondria, but does it also increase the spare capacity of individual mitochondria as well?
I really don't know. I mean, my impression is you get more mitochondria, but I don't know if individual mitochondria would respond to that, but certainly you would end up with more of them. But it's very cheap, quite fun to do as long as it's not raining.
I think the confusing thing when we talk about these physiologic manipulations is that exercise is changing your mitochondria in muscles, endothelial cells, the vascular system, the heart. And then, you know, by the time it's doing something to the brain, you know, who knows if it's related to the myriad of other changes.
Hang on, you guess if somebody's getting feedback here. I don't know. Yeah, carry on, Neil.
Oh, sorry, my daughter's on a Zoom next to me, so, I'll leave it at that. But yeah, I think that the physiologic manipulation of exercise is far removed from the neuron, so it's hard to say.
Well, no, it's removed from the neuron, but it's not removed from blood supply. And so the neuron is being killed by a lack of blood supply, and exercise seems to increase global blood supply or blood supply capacity. That would be the argument.
Yeah, no, I think if you make the argument through the vascular system, then it makes more sense.
I have a question.
Yeah. Okay, Manish.
Yeah. So my question is, like, Parkinson's disease, Alzheimer's is a long-term etiology. It takes years and years and years.
Yes.
So like low dose, like chronic exposure to stress, like say sub-micromolar sublethal dose of calcium, consistent calcium exposure, like calcium overload, how does that affect the citric acid cycle? Because, no, not the TCA cycle, Krebs cycle, because calcium is one of the cofactors or is important for activation of a lot of TCA cycle enzymes. So maybe consistent, not enough to kill the cell, but like consistently increase the TCA cycle and get a lot of NADH and then increase the ATP levels.
So it depends what is the rate restricting set of processes. If you have a situation where what is ultimately limiting is the supply of NADH to the mitochondrion through the citric acid cycle, then it would appear that if you could increase the activity of the citric acid cycle, then you would move the maximum limit up somewhat further, so it could be beneficial. I don't know of experiments that have actually tested that or whether there's any sort of evidence, but anywhere, you see, any of the upstream steps could become rate restricting, and anywhere that could show flux control is the official term, you can activate and increase the maximal capacity. And we're going to see when we do the my seahorse experiments later on, you're going to see some rather interesting and surprising effects that relate to altered upstream activity and in terms of the respiratory capacity. Daniel.
Hey, yeah, on this discussion of respiratory capacity, it's really interesting that you're seeing this age-dependent difference in...
We're not seeing it, it's purely a hypothesis. And someone is very involved in aging, because I'm doing it myself. It is a purely hypothetical.
Okay. Well, then just to add some data points to, what you're working from, I work in the retina and we routinely measure oxygen consumption. We can measure things like spare respiratory capacity, and we have and others have. And in the retina, there is absolutely no spare respiratory capacity.
So the retina is always working flat out?
FCCP doesn't alter oxygen consumption, at least not with glucose as a substrate, and the retina does love glucose.
Right. But I mean, that would be in culture, right? Or
yeah, I mean, we have like a flow culture system similar to what David described for his cells earlier on.
What I mean is, how do you correlate that with what's happening? I mean, could that be an in vitro artifact, and how do you correlate that with what's happening in vivo? Just a thought.
Yeah, I'm actually not correlating it with in vivo necessarily. I'm correlating it with David's in vitro experiments where he sees that his.
So one thing, Daniel, which will actually come up in today's seahorse experiments is whether you have the optimal substrate supply, whether glucose is optimal or whether other substrates will be better, whether there's any hormonal effect, any insulin facilitated glucose uptake or anything like that. And that's something we can sort of talk about this afternoon or later today in the problem solving, whatever it is. I get confused.
It's an important question for sure. But I did want to emphasize a lot of data to support that the retina loves glucose.
Okay, Shona.
I would jump in though and add that experimentally, depending on the way you run the experiment, you can either elicit a maximum respiratory rate that's higher than your substrate induced rate, or not, depending on how long oligomycin has been in a respiratory control experiment. So especially for cells running on glucose only, we often see that there's no apparent spare respiratory capacity if they've poked along without oxidative ATP production for a while. But if we add FCCP and oligomycin together, you don't allow the cells to go into energetic collapse, and then you do elicit a spare respiratory capacity. So the definition of spare respiratory capacity is very condition dependent and very experiment dependent.
And this is something I'm very aware of because I've been doing a lot of work with insulin secreting beta cells and beta cell lines. And what's critical about a beta cell is that it lacks lactic dehydrogenase, so it cannot do anaerobic glycolysis. So you do a classical seahorse experiment, you add oligomycin, and the cell is immediately in an energetic crisis because it can't compensate for the lack of oxidative phosphorylation by increasing glycolysis, because it's nowhere to go. The pyruvate has to go into the mitochondria. And what happens when your ATP levels start to fall, as shown as sort of discussed, is that glycolysis fails. Glycolysis has two steps that need ATP, hexokinase and phosphofructokinase, and they fail. So a collapse in ATP actually deprives the mitochondria of their substrate, of their pyruvate that they need in the presence of glucose.
Yeah, I mean, I think I just want to say that the retina is very glycolytic. I don't know that it undergoes an energetic crisis, and it these experiments were performed with oligomycin in addition to FCCP, so ATP synthase should not be running in reverse. But also, I mean, if you're interested, I can send you papers. If you're not interested, I can just not send you papers.
I'm always interested. The way in which we modified the seahorse experiment, I think somebody actually mentioned this, was adding oligomycin and FCCP together rather than sequentially. So you do one experiment with just oligomycin, another experiment with oligomycin and FCCP simultaneously, and then you get the maximal restoration before the ATP has had time to collapse.
I have one quick question, and that is, how long will the cell, I know it probably depends on the cells, and mitochondria, isolated cells and isolated mitochondria, if you provide them with substrate, how long will they last? Days, hours, days, weeks?
Mitochondria will last hours. Cells will last weeks if you have good cell culture conditions and you keep them sterile. That's one of the one of the advantages. I can, in theory, because the seahorse is non-invasive, in theory, one could carefully take the cells out, do a seahorse experiment, put them back again if you could keep sterility and look at them again. But I mean, certainly cultured cells, of course, people culture them for weeks.
And if the cells come from old rats versus young rats, or the mitochondria, does that make a difference?
What makes the difference, and which is one of the big bugbears when I was working with cultured neurons, is that cultured neurons have to come from neonatal rats. And that is the problem. I mean, one of the big limitations is that nobody, to my knowledge, succeeded in making really intact neurons from adult animals. So that's one of the big problems. I'm getting signals from Ricky here. Hello, Ricky. That we should be thinking about going into our breakout now because we don't want to run behind time. So, Martin, Shona, should we go into breakout?
Okay, welcome back, everybody. I'm going to give you a lecture now about mitochondrial production of superoxide and hydrogen peroxide, and that's what I work on and what I get excited about. What I hope this is an example of starting from understanding and playing around with properties of the electron transport chain in mitochondria, in this case in terms of the rules that govern whether a particular site in the electron transport chain is able to spill electrons onto oxygen and make superoxide. And then the development of that underlying bioenergetics and understanding into more complicated physiological models, into preclinical models, and this is the hope bit, eventually into the clinic, so that we can actually treat patients for diseases based on this bioenergetic understanding. And I suspect that that motivation is also often a motivation for you in the audience, that you're trying to do something along that same spectrum. Often people come in at the middle and they're trying to go towards the desirable outcome and they need to backfill the mechanism. In my case, I kind of started with the mechanisms and progressed in a relatively linear fashion through to the point of having things that we might be able to use in the clinic. So the structure of the lecture is going to be that I'm going to talk about the things related more directly to the bioenergetics, to the different sites in the electron transport chain that can make superoxide or hydrogen peroxide. And then start moving into the more physiological questions of how do these different sites run and when do they run and how important are they relative to each other? Which ones matter and which ones are test tube artifacts but of no great physiological consequence. And then that leads into the development of small molecules which are able to prevent the superoxide production from particular sites. And then the use of those as tools to ask the question in cells as to, well, are these sites running either in a physiological context in a cell in culture or in a pathological context of a cell which is a model of some disease. And then driving those molecules by drug development program into drugs that we could use to treat diseases and pathologies, and to show you a couple of examples of things where that seems to happen in models as a guide to where we may be able to get to in the future. And the general model that this is all built around is the idea that when you are an organism that uses metabolic reactions, in particular oxidative and redox reactions, then you inevitably generate free radical oxidative stress. And that it's wonderful to use oxygen and the big free energy that you get from oxidizing reduced carbon substrates. But the price you pay is that these radicals gradually degrade you, the biological system. And they lead to all of these nasty things, which in the end, usually post-reproductive, kill us off and limit our age span. So the free radical hypothesis suggests that a primary driver of aging itself is this radical production. In other words, the machinery we've been talking about in terms of OXPHOS in cells and in mitochondria is also the machinery that kills us in the long run by causing aging and the diseases of aging. And that's obviously a fairly controversial hypothesis. The evidence is not good that it's the only cause of aging, but the evidence that it's involved in some way, in particular involved in some of the diseases of aging, is pretty good. So I think most people would agree that oxidative stress is a factor in disease and perhaps in aging. So to help conceptualize what's going on, I've found it useful to redraw the electron transport chain in a slightly different way, but it's strongly related to the ways that David's models that we've been using up to now also organize the system. What I've done primarily here that's different is to arrange everything on the redox scale, on the EH scale. So I took these numbers from one of David's bioenergetics books, so they ought to correspond to the ones he was using, but I noticed they're just slightly different. What it represents is there's a set of carriers which are running at the most negative redox potential at an EH of about minus 280, 300. And those are all centered around the NADH-NAD pool in the mitochondrial matrix. And they include all of the Krebs cycle dehydrogenases, the pyruvate dehydrogenase complex, the dehydrogenase complex, and others. And they include complex 1 and the flavin site in complex 1 that accepts those electrons from NADH and hands them on down the chain. And then complex 1 is drawn here as a big squidgy thing with dropping in energy from this isopotential group at about minus 280 down to the next isopotential group at around 0 or plus 20 millivolts of redox potential. So this is supposed to represent some sort of water flowing down from one level to the next and a turbine, which would be the proton pumping activity of complex 1, pulling out energy as protomotive force, which can then be used for ATP synthesis. And then once you reach the redox level of the Q pool, again, there are dehydrogenases that can feed in there, in particular succinate dehydrogenase, but also the glycerol phosphate dehydrogenase, the ETF system, and some other minor Q-linked dehydrogenases. So we've talked about that in the experiments that we went through yesterday, where succinate can feed electrons in past where rotenone inhibits, which would be at this Q site here. And then again, from the Q pool, electrons run into complex 3, another turbine that drives PMF production through proton pumping down to the cytochrome C isopotential group at about plus 300, 320, where there's a few cytochrome C-linked dehydrogenases, none of them of any great physiological flux importance in that sense, but there are, for example, sulfite dehydrogenases, which are able to reduce cytochrome C and hand the electrons in. And we used in experiments a ascorbate TMPD because that's much more active. The sulfite is really not fast enough to be useful in those experiments we were running. And then finally, the electrons enter complex 4, run down to oxygen at plus 600, plus 800. And again, there's proton pumping, and you can pull out the energy as protomotive force and use it to drive ATP and the rest of the reactions in the cells. And the way we've described it up to now is a perfect machine, in that you feed electrons in from one of these substrates like pyruvate or malate. They flow down the chain and they end up going onto oxygen and forming water. And what we've avoided saying is that, but not all of them do that. Some of them escape. And electrons, as you probably know, if you have any physics background, electrons are really quite slippery things. To say where an electron is not a very easy thing to do when you get down to the quantum mechanics of it. And electrons therefore tend to escape at various points in the chain. And that's what this lecture is going to be about, is those leaks of electrons onto oxygen to make superoxide. So quantitatively, the reaction you want to happen is a 4-electron reduction of an oxygen molecule, an O2. As David mentioned, formally, we normally talk in 2 electrons and O, notional O atoms. But really, it's a 4-electron reduction of an oxygen molecule, which is catalyzed by cytochrome oxidase. And once you've reduced it, reduce the oxygen, you protonate it, and you make 2 water molecules. And this is what oxidative phosphorylation is trying to do. It's really good, and it happens nearly all of the time. So only about 0.1% or less of the oxygens, or of the electrons, escape prematurely and react with oxygen. And then when they do escape, there's basically 2 ways that they can reduce oxygen. One is via 2-electron reduction to make H2O2. Again, you protonate, so it ends up as a neutral molecule. And that's usually bad. H2O2 is a relatively reactive molecule. It can diffuse quite a long way. So you probably would rather not be doing that. But what biology seems to have done is kind of accept that it's happening and then use the H2O2 quite often as a signaling molecule. So H2O2 can often be a signal to the rest of the cell of redox events going on in the mitochondria. And then the thing you don't want to happen, that does happen, is that sometimes you can pick up single electrons from semiquinones, from iron sulfur centers, perhaps from flavins, and reduce oxygen to the superoxide anion. It could protonate, but the pK is such that at physiological pH, it's mostly the anion, not the protonated form. And this is a pretty reactive molecule, so it will react with things, in particular with double bonds, to oxidize lipids. And once you oxidize your lipids, that sets going a cascade of further lipid oxidation. And that seems to be one of the main drivers of a lot of pathologies. So you don't want this to happen. And biology doesn't seem to use it for anything. So it's just an unfortunate side effect of running electron flow down from a reduced donor to the oxidized acceptor, to oxygen. So the way I got into this problem is now about 20 years ago, and we joined a field which was trying to understand the properties of each of the individual sites. And the sites that were identified early were sorry, this one here in complex 3. So the outer Q binding site in complex 3. So that's a big mouthful, so we call it 3QO for the outer Q binding site. Seems to be a primary site in the electron transport chain that can spill electrons. And that's because there's a semiquinone formed in there during the Q cycle reaction of complex 3. And that semiquinone, if it hangs around, can donate its electrons to oxygen to make superoxide. And then following that, people discovered this site in complex 1, the flavin site in complex 1, which we call 1F, which is also able in the test tube to make a lot of superoxide. Although since then it's become clear that a lot of the things that were assigned to that site in complex 1 were actually dehydrogenases in the Krebs cycle, which were spilling electrons, and they were all lumped together as complex 1, but actually mechanistically most of that signal is probably not complex 1. And then subsequently, we and others investigated all the different sites in the electron transport chain, and each of these red dots is a site that has been shown to generate superoxide and or hydrogen peroxide. Often it's quite difficult to tell which it is because we're measuring hydrogen peroxide at the end of the day, and it's difficult to know if that's made directly or whether it's superoxide that's made first and then it's converted into hydrogen peroxide. So there are at the moment about 11 different sites within the chain that have been identified by us and by other people, which are able to generate superoxide hydrogen peroxide. The biggest surprise was complex 2, where under the appropriate conditions, which is basically a very low succinate concentration, so this flavin site on complex 2 is exposed to the air some of the time. That can be a major generator of superoxide, and that wasn't known before we started working on it. And then the nomenclature here is we've just given the name of each red dot. The complex, so this site is complex 2, so it's 2, and it's a flavin site, not a Q binding site in complex 2, so it's site 2F. Site EF is the ETF-Q oxidoreductase flavin. Site 1Q is the quinone binding site in complex 1, and 1F is the flavin binding site in complex 1. And then one of the things that's become apparent, and most of you will probably be aware of, is that if you take isolated mitochondria and you give them a large amount of succinate so that you feed electrons into the Q pool, you can get reversed electron transport into complex 1, and that can drive a lot of superoxide production. So David walked us through that a little bit yesterday. If you put electrons into complex 2, they reduce the Q pool. That's not in itself enough to send electrons back upstream because of this thermodynamic drop, which is indicated here by the EH difference. So it's really hard to push electrons backwards if you just reduce the Q. But the Q is also oxidized through complex 3 and complex 4. You pump out protons, and once you have a large proton motive force, then the combination of a reduced Q pool and a large proton motive force is now able to reverse the equilibrium of the complex 1, bring it close to its equilibrium value, and electrons can flow back by reverse electron transport into this Q site, back into the flavin site, and back into the NAD/NADH pool in the matrix of the mitochondria. And it turns out that under those conditions, this quinone binding site in complex 1 really makes a huge amount of superoxide, maybe hydrogen peroxide. And there's a controversy I won't go into as to whether that arises from the flavin mechanistically or from the quinone binding site mechanistically. I think the evidence is pretty well overwhelming that it's from the Q site, so I'm going to assume that that's the case from here on in. But be aware that there are other people in the field who say, no, it's really coming from the flavin site. And what I call 1Q is just some funny conformation of complex 1, which activates the flavin site. So the sites I'm going to be most concentrating on are 1Q, 1F, 3QO, and 2F during this lecture, because those are the ones that seem to be quantitatively the most important in all of the different systems that have been looked at. So how do we measure what's coming out of a particular site as opposed to just what's coming out of the whole of the electron transport chain? And this whole field is plagued by methodological problems. Most of the reagents that are available are no good. So most of them are non-specific, so they react with all sorts of things, including not only superoxide, but often hydrogen peroxide as well. And often they're redox active, so they'll also be reduced by other things usually unknown, such as cytochrome C, which mean that the signal that you get from most of the probes is ambiguous as to what you're really measuring. So the ability to measure superoxide is difficult. And then on top of that, the problem with electrons is that if you do the classic sort of experiments where you inhibit a site in order to see what it's doing, for example, if you dump mixothiazole on isolated mitochondria and stop the 3QO site from making superoxide, the problem is that electrons are very squishy and they want to go somewhere. So if you block the site that they're trying to get to on complex 3, then they'll go on to other sites. And so you'll get a decrease in the superoxide production from the 3Q site that you just inhibited with mixothiazole, but you'll get also an increase in all of the other sites as the electrons back up. And that also plagues the literature, because you can find a lot of papers where people do a genetic manipulation, for example. They might either mutate or knock out the risky iron sulfur center, which is talking to the QO site, and then measure using these unreliable probes changes in a fluorescent signal, which they ascribe to superoxide or hydrogen peroxide. And that's really problematic on both the levels I've just been through. The probe isn't really specific for the superoxide, so it might be telling you about something else, such as the redox state of some other center in here. And in addition, when you knock out the risky iron sulfur center, which is a component of this Q site, then the electrons redistribute and other sites start to increase. And even worse, any sites downstream decrease. So that's perhaps better illustrated if I talk about using rotenone as the inhibitor. If you inhibit the 1Q site with rotenone during normal electron flow, not only do all of these sites upstream go faster, but all of these sites downstream get starved of electrons, so they go slower. And what you see is some resultant of some sites going faster, some sites going slower, and the site that you've blocked out. And that makes it really difficult to interpret most of the literature about the different sites of ROS production in cells and in mitochondria. So we spent a lot of time over the last about 10 years now trying to find ways in which we could overcome those problems. And we started by using endogenous reporters of the other sites. So if the problem is that other sites are going faster or slower when you inhibit a particular site, then you can compensate for that by measuring how much faster or how much slower the other sites go, and then mathematically correcting. And then when you add your inhibitor, when you add rotenone or mixothiazole, if you correct for all these other changes in the system, you can then correctly ascribe the change in superoxide production to the site that you've inhibited. And then coupled to that, you need to use a probe system that you believe in. And we trialed all of the ones that were on the market and decided none of them were any good. And the one we've ended up using is the Amplex Red horseradish peroxidase system, which is an enzymic, clunky, old-fashioned system where the horseradish peroxidase takes H2O2 and reduces a dye, which then, I forget if it's an oxidation or a reduction, it reacts with the dye, which then gives you a fluorescent signal which you can follow in time. And then, as I'll go on to develop, using those techniques, we were able to understand these sites, and we could use that understanding to develop site-specific suppressors, cycles and sequels, which is what I'll be talking about for a large part of the lecture. So how do endogenous reporters work? So an endogenous reporter is something that you can measure, for example, the NADH-NAD ratio in isolated mitochondria, which is reporting or is in equilibrium with the activity of some site that we care about. So for example, the flavin site in complex 1. So it turns out that the flavin site in complex 1 is pretty much in equilibrium with the NAD pool that it sees in the mitochondrial matrix. So if we arrange things appropriately by blocking off with rotenone anything else that's happening downstream in the electron transport chain, and by using appropriate substrates that feed into the NADH-NAD pool without feeding electrons to any of these other sites that are also talking to that pool, then we can get a clean signal which is due only to a single site, and we can relate how fast that site is going to the redox state of the NAD pool, which we can measure. So this is an example of that, where we use our Amplex red horseradish peroxidase system in isolated mitochondria under contrived conditions where we think only site 1F is running, and we measure its rate as a function of the NADH signal that we can get from the mitochondria by looking at endogenous autofluorescence. So as we mentioned yesterday, you don't really know if you're looking at NADH or NADPH, so we call it NAD brackets PH reduction. But as I mentioned in the discussion, under the conditions we use, the NADPH is pretty much fully reduced all the time and doesn't change very much. So nearly all of the signal that we see is probably the NADH in the matrix. So you can get a relationship between the amount of H2O2 that you can measure and the NADPH reduction. And that allows you to know how fast 1F is going at any particular NADH reduction in an experimental system where you don't contrive the conditions. And then after we published that, we discovered that we could take out some of the signal by blocking off some of these dehydrogenases upstream, so they were still contaminating our signal. And when we use a combination of inhibitors that remove these activities, then these cyan dots here give us the true relationship between the flavin superoxide production and the NADH-NAD ratio. So that's all done with contrived conditions with inhibitors present, so we really can understand which particular site is running. But what we can then do is use the NADH-NAD as an endogenous reporter under any other condition for how fast this site is going. So in some isolated mitochondria, given some complicated mix of substrates under some regime of ATP synthesis, we can measure the NADH-NAD ratio and then use this as a lookup. So if it's 60% reduced, we can just read off, okay, then this site must be going at 20 picomoles per minute per mg of mitochondrial protein. We also were able to use an endogenous reporter for the 3QO site, which was the cytochrome B that's sitting next to it, which you can measure by absorbance spectroscopy. And similar ways of contriving a system, so only that site is running, doing a calibration, and then using the calibration to read out under more complicated systems how fast 3QO is running allowed us to also measure the 3QO. And as I mentioned, the 3QO and the 1F are the dominant sites under many conditions. So when you poke around with, say, rotenone to inhibit the 1Q site, the major sites that change are the 1F and its upstream friends and the 3QO. So we can use the endogenous reporters to correct for those changes and then correctly measure what's going on in 1Q by allowing for these secondary changes after you add inhibitors in the upstream and the downstream sites. So that turned out to be quite hard work, but very effective, and was able, at least to our satisfaction, to solve the question of which sites are running in isolated mitochondria under different conditions of incubation. So the first thing that we did was to relate back to the field that this all developed out of, which was to try and maximize the superoxide production from any site of interest. So here what we're doing is we're trying to understand the capacity of a particular site. How fast can we go if we push it under optimal conditions as fast as we can? And so these upstream sites, all in the NADH isopotential group, we were able to use various substrates and inhibitors to differentiate how fast the oxyglutarate dehydrogenase flavin is running, site OF, the pyruvate dehydrogenase flavin, various branch chain and other oxoacid dehydrogenases. And the surprise there was that the flavin site of complex 1, which up to that time had been thought to be one of the quantitatively major sites, actually isn't that quantitatively big. Most of the signals that people get if they just add rotenone to mitochondria and pump in some substrates and measure the H2O2 production, most of that comes from oxyglutarate dehydrogenase or pyruvate dehydrogenase, depending on what substrate they're adding to the isolated mitochondria. And then this interesting reverse electron transport driven site, site 1Q, can really go faster than any of these NAD-linked sites. So when you drive reverse electron transport from succinate back up into complex 1, this is the dominant site that you see. You see the 1Q site. And I'll show you the evidence for that statement just a sec. And then around Q, there are a set of sites which are able to make superoxide. And as I mentioned, the 3QO site on complex 3 is actually a site that you can get a huge amount of superoxide out of. And that one's relatively well understood. There's a semiquinone that's formed in that site. And if you poise the redox state of that semiquinone, so it's the Q in that site is largely in the semiquinone form rather than the fully reduced or the fully oxidized form. So you optimize the conditions, you can make this thing run really fast. And that you do by adding antimycin to block this site so the electrons don't have anywhere to go. Normally, of course, they would just flow on down the chain and they wouldn't accumulate in this site. So this is a contrived condition to drive the site as hard as we can drive it. And when you do that, it's the site, at least in rat muscle mitochondria, which is our usual test model, it's the site that has the biggest capacity by a long way over the other sites. And then the ancillary dehydrogenases that feed electrons into the Q pool, complex 2, the 2F site really can go quite fast. And in these muscle mitochondria, the glycerol phosphate dehydrogenase is relatively active and is a contributor. But if you switch to other mitochondria where they use glycerol phosphate much more avidly as a substrate, such as brown adipose tissue mitochondria, which David will talk about tomorrow, then the contribution of this GQ site, the Q binding site in the glycerol phosphate dehydrogenase, can be up here equal to or greater than some of these other sites. So how large a capacity a particular site has depends on the enzymic makeup of the mitochondria that you're looking at, and it'll differ from tissue to tissue. And then one of the other interesting aspects of this is that all of these different sites make 100% of what they're making into the mitochondrial matrix, except for this 3QO site, which makes superoxide approximately equally to both sides of the membrane. And it's a little bit mysterious how it does that. There are different models of it. But operationally, what that means is that it's really only from the 3QO site, or perhaps from this glycerol phosphate site, that you can make superoxide directly to the outside of the mitochondria. So if you sit in the cytosol or in the nucleus, the only sites that make superoxide from the mitochondria that you can see would be the complex 3 site or the glycerol phosphate site. All of these other sites are making superoxide, but they're making it in the matrix. And you never see that if you sit in the nucleus, because superoxide effectively doesn't cross the mitochondrial inner membrane. Of course, there's a superoxide dismutase in the matrix that converts that superoxide into hydrogen peroxide, and the hydrogen peroxide can diffuse across the membrane. So what the rest of the cell sees is hydrogen peroxide coming out of the mitochondria, plus any superoxide that's being made from the 2 sites that partially face outwards. What the matrix sees is about half of this 3QO, so it brings it down to about here. And it sees some mix of all of these different sites making mostly superoxide, but partially hydrogen peroxide. So in the mitochondrial matrix, if you're sat there, what you see is this flood of superoxide coming out of all of these different electron transport chain sites, which are running electrons. And the question we're interested in then is, so which sites are important? Which ones give you the biggest signal and therefore may drive all of these nasty things that in the end we want to avoid? So I've been talking on conditions that approximate to Vmax for an isolated enzyme, a capacity measurement where you optimize the conditions, you drive it as fast as you can, just so you get some sense of what's going on. And that's really useful in a preliminary categorization of what do you have in this box of electron transporters in terms of which ones can do it fast and which ones can do it slow. And it's really useful for understanding mechanisms. So when you're trying to understand why is it that complex 3 makes superoxide, what's the mechanism of that, how are the electrons arriving on the semiquinone, then these Vmax conditions are really useful because you get a big signal and you can get a nice large signal to noise ratio, so you can do experiments to find out what's going on. And they're quite good for comparing the capacities of different tissues. So you can compare muscle with heart, with brain, with brown adipose tissue to get some sense of which sites matter most. And it turns out they're really helpful in running screens, which I'll come back to in a moment. And of course, if you are interested in pathologies, if you find that a particular site is super active or super inhibited in some pathological condition, that's a potential cause of the pathology. So it gives you somewhere to look when you're interested in Alzheimer's or Parkinson's or heart disease or metabolic syndrome. If a site is very changed under conditions of that pathology, maybe it's actually doing something there. But what we really care about is how fast is a site actually going. So we stumbled to find a good terminology for that, and we ended up calling it the native rate. And that's equivalent to the small v in a Michaelis-Menten analysis. It's the actual rate of the enzyme under the particular conditions of substrate and all the other effectors that you're adding. So in terms of the superoxide and hydrogen peroxide, what we're working towards the question of how fast is a site running under some particular conditions where we haven't constrained it by putting a lot of inhibitors in there. And in the end, the question we want to ask is, how fast is site 1Q running right now in your brain or in my muscle in order to either have physiological or pathological effects? So this is useful for trying to understand how the flows of electrons drive superoxide and hydrogen peroxide under different conditions with different substrates, under different physiological conditions of energy demand, and under different conditions which mimic different pathologies, such as cell models of Alzheimer's or other biologically relevant models of diseases.
Martin?
Yeah.
I have a quick question, which is, I mean, there's definitely, for any kind of velocity, there's going to be dependence on the concentrations of substrates. And the electrons are one substrate, but also oxygen is another. So I was wondering if you found sort of a low or maybe higher oxygen dependence for some of these rates that you're seeing.
Yeah, we haven't worked on that, but other people have. And after some initial confusion, it's now generally agreed, I think, that oxygen concentration is a linear component of the ROS production from any particular site until you get to very low oxygen. So if you halve the oxygen concentration, you halve the rate from all of the sites. And that seems to be true for all of the sites down to very low oxygen when you start to get some affinity effects which change that relationship. So yes, the oxygen tension matters, and most of the things I'm going to talk about, we ignore that. So I'm going to be working with isolated mitochondria under air saturation, isolated cells under air saturation. But in the end, what we care about is the rate in a real organ, in a real animal under non-saturating oxygen conditions. So yes, it's a variable. The general idea is that all of the sites will go up and down in concert when the oxygen changes. So any conclusions we make about the relative importance of different sites remain true as the oxygen goes up and down, but we get the absolute rate wrong when we try and do that. And as you know, oxygen tensions are very labile and relatively difficult to measure. So we're trying to keep things simpler rather than including them right from the start as a variable. Okay. Okay, so we're now shifting from the optimized conditions for all of the different sites to the standard mitochondrial experiments of the sort that we were going through yesterday, where you take isolated mitochondria, you give them succinate as a substrate, for example, and then you ask, or we asked, which sites are running if you just give mitochondria succinate? And we can ask, and we'll ask in just a moment, the same question, what if you give them glutamate and malate or fatty acids as a substrate? And this is a stepping stone to the question we really want to get to, which is what are mitochondria doing when they see a real mix of substrates that they would see when they sit inside a cell. But the whole field is built around isolated mitochondria being driven by artificial saturating substrates. So that's where we started. So we can measure the amplex red signal using this horseradish peroxidase system under conditions of saturating succinate concentration, and we get a rate of superoxide hydrogen peroxide production. And then using these endogenous reporters, we can measure the NADH-NAD ratio and ask how fast is this flavin site on complex 1 going. And it turns out to be a really minor contributor to the total rate that we see with succinate. Similarly, we can use the cytochrome B as the endogenous reporter to tell us about 3QO and how fast that's running. And again, it's really not that important under these conditions. And the site that's really running, as everybody already knew, and as you know, is this reverse electron site, site 1Q, under those conditions. So the reason 1F is running is that the electrons are going up into complex 1 and coming onto 1F, and there's a small rate from 1F. But it's the stalling of the electrons in 1Q that really causes the maximum, the largest component of the superoxide production. So to a first approximation, if you add succinate to isolated mitochondria, and this turns out to be true for just about any mitochondria people have looked at, then the major driver of the superoxide and the hydrogen peroxide that you see is the 1Q site running under reverse electron transport conditions. And then we can do the same thing with different substrates if we use glycerol phosphate or palmitoyl carnitine as a representative fatty acid oxidation substrate, or NAD-linked substrates such as glutamate and malate, then we can repeat the same analysis. And what you can see is that when we use glycerol phosphate, it's also able to drive reverse electron transport into complex 1 in the same way as succinate. But now the glycerol phosphate dehydrogenase itself is also a major contributor. And to our surprise, under these conditions, the complex 2 flavin now starts to make superoxide. So when we have succinate as a substrate, the succinate binds to the active site of complex 2 and blocks the access of oxygen. But if we use glycerol phosphate as a substrate in the absence of succinate, that site is now open and available. So electrons flow from the Q pool back into succinate dehydrogenase onto the flavin of the SDH, and then they react with oxygen to make superoxide. So we see a 2F component under those conditions. And 1F and 3QO are still running. And again, differences with other mixtures of substrates. So what we can say from these isolated mitochondrial experiments is that the rate of superoxide production from the electron transport chain depends really quite strongly on what substrates you're giving the mitochondria. And that maybe was obvious, but this is a nice demonstration of it. And then something else, which in retrospect is very obvious, but wasn't so clear before we did it, was that the sites that are contributing are also rather different or very different, depending on which substrates you're oxidizing. And in particular, if you're oxidizing succinate or glycerol phosphate, you get a lot of 1Q superoxide. But if you're oxidizing fatty acids or NAD substrates, you don't get any. So the contributions of different sites vary depending on which substrates you're oxidizing. So what we want to do then is to ask, well, what if you give mitochondria what they would actually oxidize in a cell? And we wanted this as a stepping stone to the question of what's happening in real cells. But because we didn't at that time trust any of the probes that people use in cells, we had no tools to measure the superoxide production, and therefore we couldn't do these experiments in cells. So instead what we did was to take isolated mitochondria and then look up the extensive 1970s literature about what are the substrate concentrations for mitochondria in intact muscle under different conditions of rest and exercise. And then we went through everything that the literature said, discarded all those things we thought weren't important, hunted down everything we thought should be measured and should be important, and came up with a complicated cocktail which represents the substrates that muscle mitochondria interact with C when that muscle is at rest and when it's under conditions of exercise. And then we went back and we gave our isolated muscle mitochondria that mix of substrates that we hope represents what they would see in vivo, and asked how fast does superoxide hydrogen peroxide run under that condition. So I've now plotted that on the same scale at rest and exercise. And what you can see is it's really only a very small proportion of the capacities of all of these sites. So these sites have a huge capacity to make superoxide hydrogen peroxide, but under plausibly physiological conditions, they're just not utilizing most of that capacity. So these are now not optimized for superoxide production, and under more natural conditions, the sites are ticking over, but they're not going that fast. So we can't a priori say, just because this site happens to be the one with the biggest capacity, that it's an important site in muscle at rest. It might be one of these minor sites, like the flavin site in complex 1, is more important in muscle at rest. And then the other thing we saw, which is to a bioenergeticist completely natural and what you would expect, is that if you mimic exercise by having the right substrate combinations for exercise and by turning over ATP at something approaching the state 3 rate, then the superoxide production by the electron transport chain goes down. And that contradicts what the exercise physiologists know, which is if you exercise, your ROS burden in muscle goes up. So what we conclude from that is the signal that you see in muscle, either we've just wrongly worked out what the metabolites are in muscle, or there's other signals that matter in this system that we don't know about. Or the one we think is the more likely is that actually it's not the mitochondria that are making this ROS that you see under exercise in muscle. It's other sites, in particular the NADH oxidases in the cytosol and on the plasma membrane, which are able to make superoxide under stress. And we think those are the ones that are occurring during exercise. The reason that the mitochondriac thinks that you get a lower rate during exercise is that exercise is akin to state 3. And if you run mitochondria in state 3, the electron transport chain goes oxidized because you're draining out electrons in order to pump the protons that you need because of the non-equilibrium that we've set up in order to drive this state 3 condition. And if the redox centers in the electron transport chain go more oxidized, they have less electrons on them to donate to oxygen to make superoxide. So you'd predict that they would go slower. And that's in fact what we see in this experiment, is most of the sites are oxidized and go slower than they do at rest. So in the mitochondrial perspective, the dangerous thing to do is to sit quietly at your chair listening to a lecture, because your mitochondria are making about twice as much ROS as they would be if you're exercising and keeping all of your redox centers oxidized. But that needs to be offset by the rest of the cell, which is making superoxide.
Martin, I had a question. Sorry, I missed the part of, so the left is all set up artificially, I understand. How do you take the mitochondria resting versus exercise? How do you do that experiment?
So this is done by taking out of the literature the concentrations of all of the amino acids, Krebs cycle intermediates, lipid species, and ADP, phosphate, calcium, magnesium, pH, that people find at rest, and dumping our mitochondria in that and saying this is like a muscle at rest. For the exercise, we use the mix of metabolites you see during exercise, and we also put the mitochondria into something close to state 3 by using a hectokinase system to recycle the ADP, the ATP back to ADP, so we can keep them in a steady state close to state 3 but not quite there. So it's an artificial mimic of exercise and rest.
And there would not be an in vivo way to measure this, right?
I'm coming on to that. So there are ways which we develop based on this, but it's quite hard to do it because the probes for measuring the superoxide are not reliable.
Thank you.
Okay, so then we're able to analyze these conditions in the mimicked rest and exercise, and using the same endogenous reporter methods. And what we find is that under rest, it's only 4 or maybe 5 of the sites that make a significant contribution. We found no contribution of these other sites, or within the noise, sometimes it was negative. And there are 4 sites that seem to be about equal: the 1Q, the 1F, the 2F, and the 3QO. We found a contribution of the fatty acid oxidation EF site, but that's got a big error bar in it, and that wasn't very reproducible, so we think that was just a part of the noise in this system. And then under exercise, everything goes more oxidized, so it goes more slowly, so all of these sites collapse into the noise. The only one that stays the same is the NADH linked, the 1F site, and that's because the biology is set up to maintain the NADH reduction state. So when you exercise, calcium goes into the muscle, that activates a bunch of things, including the glycolytic flux, and that maintains the NADH in the mitochondria at about the same reduction potential as it was at rest. So 1F, which is talking to that NADH pool, goes at about the same rate. So in the end, what happens during exercise is everybody collapses except for 1F, which is kept going. And 1F was a very low capacity site, but is the single dominant site, we think, in muscle at rest, at least from these mimic experiments. So to recap that, only 4 of these sites really seem to go in muscle under conditions that mimic rest. And similar things have been found in heart and to some extent in liver. So it may be general that these are the sites we really need to care about, and these other sites are possibly sites that can be engaged under particular unusual conditions, but they're not generally running. So it's those 4 sites then, or some subset of those 4 sites, that would be the output from life that's causing all these nasty things. So those are the sites that we should really concentrate on. And then, as I mentioned, one of the big advantages of having the ability to measure the maximum capacities is we were able to set up a screen for this rather improbable hypothesis that there exist in the universe of chemicals molecules which can specifically prevent a site from making superoxide or hydrogen peroxide without preventing it from doing the day job of running oxidative phosphorylation. So we set up a screen where we measured the X02 production with the horseradish peroxidase Amplex Red assay. And we set it up under conditions where only a single site was running, either the 1Q or the 1F or the 3QO or the 2F or the GQ site. And we ran a counter screen where we did a bioenergetic screen. We initially used the membrane potential of the screen because it was a convenient fluorescent assay. And we discarded anything which was able to interfere with the energetics. And we also discarded anything which was able to hit more than one site on the grounds that non-specific antioxidants would hit all of the sites. So we can eliminate them just by removing anything that hits more than one site. And what we found is, surprisingly, that there are quite a lot of molecules in that chemical space which are able to prevent superoxide production from either site 1Q or site 3QO without affecting the bioenergetics in any way that we can measure. So initially we did 3,500. On the basis of that, we then partnered with Novartis in San Diego, and they screened for us 635,000. And on the basis of that, we're still running screens, and we've done more than 1 million to date to find compounds which are successful at preventing superoxide production and also have other desirable properties that make them drug-like. So here's an example of the complex 3 suppressors. So we call them suppressors of site 3Q electron leak, S3QEL, and then we call that sequel. It's kind of clunky as a name, but that's what we did, so we're stuck with it. So sequels are compounds which have these structures, the ones we found in the first screen, quite unrelated chemical structures. Compounds which can take out the activity of the 3QO site without affecting any other site and without doing anything to the bioenergetics, without doing anything that stops electron flow or oxidative phosphorylation. And if we find any of the subset of molecules we're working with which does something, we throw it out. So we keep a clean set which, under the conditions we're using, doesn't do anything to oxidative phosphorylation. This is the seahorse experiment just showing that. So for sequels, we're measuring state 2, state 3, state 4, O. So we're measuring the effect on the proton leak, on the HB synthase, on the electron transport chain, and the different bars of different colors are just different concentrations or different specific sequels. And they don't do anything with any combination of substrates that we've looked at. So they're clean bioenergy. And these are the cycles, the 1Q, suppressors of electron leak. Same thing, we can specifically take out the 1Q site with here 2 different families of molecule without doing anything to other sites. And I won't show it, but without doing anything to the bioenergetics. So this is wonderful. We now have tools where we can go into any biological system. We can specifically prevent site 1Q from making superoxide without, if we're lucky and if we're right, doing anything else to the biology at all. So we can ask, what's that site doing in isolated mitochondria? What's it doing in a cell? What's it doing in a cell model of Alzheimer's pathology? What's it doing in a mouse model of a particular disease? And if it's doing something, if we take it out, is that then preventative or curative of that disease condition or that pathology? And we can do that with cycles, we can do that for site 3QO with the sequels. So these are really exciting tools to work with. and in principle could lead us to molecules that we could turn into drugs, and we could treat any diseases that might be driven by reverse electron transport 1q ROS production or by 3q ROS production in vivo. So we can use them as tools, first of all, to answer the question that Pankaj raised, which is what's going on in a real cell. So in a real cell, what's happening is, this is just an INS1 cell, just for illustration, where the red threads of the mitochondria are stained with a suitable stain. The green is just outlining the cytosol. And all of these different compartments, the mitochondria, the ER, the Golgi, the plasma membrane, other membranes, are making superoxide and hydrogen peroxide and dumping it into the cytosol of the cell. If any of these are making superoxide, there's plenty of superoxide dismutases around, so they'll pretty quickly turn it into H2O2. So there'll be a low concentration of superoxide, but everything will eventually end up as H2O2. And then most of that H2O2, as David mentioned, is being mopped up by the glutathione system and other antioxidant defense systems in the cytosol. But some of it escapes those and diffuses out of the cell as H2O2 leaking to the medium. And that we can measure reliably. So if we add horseradish peroxidase and Amplex red, then we can get a fluorescent product and we can measure that. And because it's enzymically driven, it's very specific for H2O2, and we can do controls to show that if we take out the H2O2, we take out the signal. So we know we're measuring H2O2 leaking out of the cell. And then we can use these tools we've just developed. We can use the cycles, the sequels, and inhibitors of NOXes, NADH oxidases that produce superoxide, to ask, okay, how much of this stuff that's leaking out can we get rid of if we just add a cycle? And that will tell us how fast site 1q is running in these cells as a proportion of the total things that are making H2O2 in the cytosol, which we're monitoring by the small fraction that leaks out. Same with the sequels. How much can we take out the sequels? How much can we take out with inhibitors of the NOXes? So this shows the experiment published last year where we titrate out with a NOX inhibitor, and we can take out in these particular cells, which are C2C12 myoblasts, about 45% of the total H2O2. So a lot of the signal we're seeing is coming from the NADH oxidases sitting in the plasma membrane or in the ER membrane or wherever they are. Two different NOX inhibitors do exactly the same thing, so that's a specificity confirmation. If we use sequels, it's pretty noisy, but we take out consistently about 15%, cycles, about 15% of the signal. And if we use sequels, we can take out about 35%, 30%, using, again, 3 different sequels with different structures, so we can be fairly confident it's on target. And you can see we get some off-target effects which start to screw things up. So it's a relatively difficult, noisy titration, but the fact that 3 different molecules will give us about the same answer gives us some confidence that that's the right answer. So we can just express that as a stacked bar. This is the NOX contribution, about 40%, 35%. The 3QO contribution, about, I forget, 20% was it? And the 1Q contribution, about 15%. And if we mix all the inhibitors together and add them, we get about the same answer. So these sites are not talking to each other. If we inhibit one site, it doesn't affect what the other sites are doing. Thus, we get the same effect if we add them all together as if we add them separately and take out sites one by one. So that tells us 2 things. It tells us that in these resting C2C12 cells, the mitochondria, as represented by site 3q and site 1q, are a major contribution to the H2O2 that's in the cytosol and that leaks out to our detection system. And so are the NOXes. The NOXes in the membranes are also major contributors. So that answers one of the questions that was problematic in the literature, which is, do mitochondria actually contribute anything to the ROS burden in the cell? The answer is yes, they do, at least in these C2C12 cells. And then secondly, which sites are doing it? And you can see that 2 of the 4 sites that we think are important are the major contributors. There's an unidentified proportion, we don't know what that is. It tends not to respond to things that we do to the mitochondria, so we suspect it's cytosolic, but we don't really know what it is. So that's the unidentified. And then what we've done more recently, published this year, is just survey a range of different cells to see if that pattern is typical or atypical. Again, we see the same thing in all of the cells we've looked at. So in purple here is the NOx, and it's always a largish, probably the biggest single component. In green is the 3QO, and it's always there, and it's always a significant component. And here's the 1Q component, same thing, it's always there, it goes up and down a bit, but it's always a 15-20% of the total. So those conclusions from the C2, C12 myoblasts seem to be true for all the cells that we laid our hands on and tested. And we chose cells from different tissues, from different cancerous states, from different organisms, and they all seem to behave essentially the same in terms of the proportion of the different sites, even though the absolute rates are very different. And then if you sit yourself in the mitochondrial matrix in these cells, you don't care about what the NOxes are doing, because that's making superoxide and H2O2 outside the matrix, and you never see the superoxide, and you probably don't see much of the H2O2. So the only thing in terms of superoxide that you care about in the matrix is the sites that are in the mitochondrial electron transport chain. And you only see half of the complex 3 because half of it goes to the outside. So if we take half of the green bar and the whole of the pink bar and then express that as 100%, and then ask what's happening in the matrix, then the answer is that pretty much 2/3 of what you see is coming from a single site, this reverse electron transport driven 1Q site. So we assert that that's the dominant site of superoxide production in the mitochondrial matrix, which is driving all of these pathologies and maybe driving aging. A single site is the one we really need to worry about. And then 3QO is also contributing, so we need to worry about that too. And the unidentified, maybe some other sites are running a bit, but they're not as dominant as the 1Q site. Even if this is all a single site, only in 1 or 2 cells does it approach the rate of the 1Q. If we assume all of the unidentified is actually in the matrix, we can add that in, and so still 1Q is dominant and 3QO is important, but the unidentified might also be important depending on what it is. So from that, we conclude that 1Q is the site we really need to hone in on in order to understand physiology and pathology of ROS production in mitochondria. And we can therefore ask what's happening in physiology, and that's what we're doing at the moment under different conditions of different substrates or different energy demands. How does that site behave? And we can also obviously look at what's that site doing in pathologies, or does some other site come up in a particular pathology? So this question can now boil down to site 1Q and site 3QO are what connects life to all of these nasty things, if that's indeed the main driver of those nasty things. So how do they compare to traditional antioxidants? So here's site 1Q sitting in the matrix. It's making superoxide, which will do things in the matrix. It will inhibit various enzymes. It will modify thiol groups. That superoxide is converted into H2O2 by superoxide dismutases, and the H2O2 can also do things in the matrix, in particular lipid peroxidation reactions, which I've shown as a separate arrow here. And then it can also diffuse into the cytosol, so what the rest of the cell knows about is the H2O2 that's being made indirectly from 1Q through the sol that's converting it to H2O2. And then the lipid peroxidation is a product of mostly the H2O2, a little bit of superoxide. And that lipid peroxidation itself sets off a chain reaction of more lipid peroxidation, and that can dominate some of the nasty things that go on. So it's what happens downstream of the superoxide production from the 1Q site. And the sort of antioxidants that we have available are general antioxidants of unspecified specificity, which do something. And it's unclear where they do it, when they do it, what they do. So if you're lucky, a so-called antioxidant might hit something that matters in this system, but it might hit something in some other membrane. It might hit a nitrogen-containing molecule, not one of these oxygen-containing molecules. So sometimes antioxidants may work and sometimes they won't. It depends on what they are and how they work. More useful are things like MitoQ and SS31, which can directly impinge on the lipid peroxidation reactions and take those out. Mitotempo is also a SOD mimetic, so it can take out the H2O2 and some of the superoxide by also removing superoxide. So Mitotempo, which is a mitochondrially targeted spin trap which interacts with superoxide and hydrogen peroxide, is quite a good way to clear the superoxide and the H2O2 in the matrix. And then various genetic manipulations, particularly a mitochondrial targeted catalase, are a good way to get rid of matrix H2O2. They're good proofs of principle for how you might want to treat a disease, but expressing catalase in the mitochondrial matrix is not a viable therapeutic option at the moment for treating Alzheimer's disease. And then what a lot of the field does is to use antimycin or mixothiazole or rotenone and try and understand or suppress what's going on with 1Q, or add even worse mutations like changing the risky iron sulfur center to affect what's going on, in that case, in 3QO. And these are absolutely horrible things to do, and they will affect the bioenergetics. And I would never go near them with a barge pole, because these are going to affect the bioenergetics. And once you start screwing around with the bioenergetics, you don't have a cure, but you have a bigger problem than you had before. So don't use poisons to treat Alzheimer's disease. Some of these things might work, but hopefully, if Alzheimer's does indeed have a 1Q component driving it, hopefully cycles would be a really effective way, because they choke off the supply of superoxide. So none of these things happen because nothing's getting into the system to cause the problem. So we have great hopes that cycles and sequels and other suppressors of ROS production, to the extent that any of those sites are driving pathologies, then the cycles and sequels should be a wonderful way to treat diseases caused by those things. I'll just run on for about another 5 minutes, if that's okay. So I've spent a lot of time digging into the literature to try and understand the literature about mitochondrial ROS production and what it does in terms of signaling and disease. And I developed a bunch of criteria, which I then applied to the vast literature and put out a review article, which is in press, which will be coming out in critical reviews in biochemistry and molecular biology, which will come out shortly. And I think there's good evidence that all of these different, roughly called signaling pathways, have a component or a strong component of mitochondrial superoxide hydrogen peroxide production. So cellular senescence, which is important in aging, differentiation, ER stress propagation, hypoxia response, the immune response, autophagy, apoptosis, cell cycle, all of these things use mitochondrial superoxide or hydrogen peroxide as one of their, usually as one of their drivers. So we will be interfering with those with potential drugs, and that's something we need to be aware of. But we also have a good tool for understanding the role of mitochondria in this process. And then the list of pathologies is even bigger. There's just about any pathology you can find papers and reviews that suggest that mitochondrial ROS may be important. If you subject that literature to a critical appraisal of how good's the evidence, many things still come through. So these are just alphabetical, but all of your favorite diseases are in here. So all of your favorite diseases have good evidence from the use of Mitotempo or the use of mitochondrially targeted H2O catalases or the uses of MitoQ, that there's a mitochondrial involvement through oxidative stress in driving that particular pathology. And all of the pathologies that are related to aging are in this group. So there's a reasonable chance that we can do things to aging-related diseases and possibly to aging itself, to the extent that's driven by oxidative stress rather than by the other known pillars of aging that people talk about. So where we're going at the moment is to try and understand how we can develop cycles and sequels into drugs to treat all these diseases. And I just want to show you some of the published examples of that, where we can interfere with various signaling processes such as the HIF1 signaling in hypoxia or caspase activation after ER stress. And we can interfere with various pathologies such as ischemia reperfusion injury in heart or the function of pancreatic beta cells if we damage them by an overt oxidative stress. I'll skip that. So here's the example of ischemia reperfusion injury, which we published now several years ago with Paul Brooks's lab. So here what you do is you take a heart in a Langendorf perfusion, you hang it in the perfusion, and it can beat for a long time regularly. But if you shut off the perfusate so it goes hypoxic, then it stops beating and you get a stalled heart. And during that process, you get an ischemia reperfusion injury. So when you go ischemic, that's in itself not particularly harmful. The heart just stops beating. But when you reperfuse, there's a huge burst of oxidative stress, and you get a reperfusion injury, which then leads in the real world to cardiac pathologies and heart attacks. In the model, we can demonstrate that by measuring the rate pressure product, which is an index of the functionality of the heart. It starts here set at 100%. When we shut off the flow of perfusate, it drops, the heart stops beating, so it drops down to 0. And then when you reperfuse in the controls, it comes back up, but it's now damaged by that reperfusion injury. So you only get back to 30 or 40% of the functionality that you had before. So that's a demonstration of the reperfusion injury in the isolated Langendorf heart model. If during the green bar only we have a cycle present at about micromolar, then we get, and then take it away again after the 10-minute reperfusion time, then we get almost full protection. With this particular experiment, it was about 80% protection. With other compounds we've run, we get back up to 100 or even 110% of the control. So the model for what's going on is a classic model now, which is developed by other labs. So we just apply it to explain our experiment. And that is that during ischemia, succinate builds up, probably just because the Krebs cycle isn't running, and various things are feeding carbon in, and it ends up sitting on succinate. So during this ischemic period, here succinate builds up. And then when you reperfuse, suddenly it's just like the isolated mitochondria given succinate. You run reverse electron transport, you run site 1Q, and you make superoxide and H2O2, and that superoxide H2O2 causes the injury, which damages the response of the heart. And if we have cycles on board just during the reperfusion period, you can burn off the succinate because succinate oxidation is fine, but you don't make the superoxide, so you don't get the injury, and therefore you get prevention. So there are other ways of trying to do that, and other groups are inhibiting succinate dehydrogenase during the reperfusion, which does seem to work and tends to support the underlying theory of the model. But I wouldn't really want my Krebs cycle to be shut down by anybody, so I would rather use cycles in principle than use SDH inhibitors as a method for controlling the ischemic reperfusion damage in heart. And then finally, I'll finish up with the effects of sequels on gut, diet-induced gut permeability. This is in press, it hasn't come out yet, but it should be out shortly. So the idea here is that during various oxidative insults, including aging, the gut epithelial lining becomes permeable, and that allows nasty things from your gut to enter your bloodstream, and that causes damage, probably by immunological effects. And we can assay this in flies or in mice. So we did this in collaboration with Pankaj, who's a member of the audience. So this illustrates the experiment in one direction, where we feed a dextran that has a fluorescent tag on it, so we can find it again after the experiment, feed it to mice, and it's able to cross a damaged epithelium and appear in the blood. So if we take blood samples, we can see how much permeability of the gut there is to this material coming in from the contents of the gut. And this is the time after being on a high-fat diet, which is an insult to mice. In the control, you get a slow increase, so normally as the mice age, then their gut becomes more permeable. But in the presence of the high-fat diet, you get a much greater permeability of the gut. So the gut's being damaged, and that may be one of the drivers of the various syndromes that high-fat diets cause. But if we have a cycle present, fed to the mouse, or IP injected into the mouse, I forget which way we did it in this experiment, I think it's feeding. then we can completely suppress that response to the high-fat diet. So this shows the raw data. This shows the area under the curve of that data. We get the increase in the high-fat diet, and we can completely prevent it by having the sequels on board to prevent superoxide production from complex 3 somewhere in this system. And we have indirect evidence to suggest it's in these cells themselves rather than somewhere else in the system that it's acting. And we can also do the inverse experiment. We can measure the leakage of albumin from the blood into the gut, again through the same pathways of the permeabilized gut. And there it's even more effective. So here's the control, in gray, here's the control. Here's the permeabilized gut after the high-fat diet. And when the sequels are on board, we drop below the control and things are even better than in the control. So sequels are able to prevent this gut permeability induced by an insult in mice and in flies. I'm not showing that, suggesting that they may be drugs that we could use to achieve that end in humans, and that would be somewhere we would be driving to try and find a home for the use of our compounds. So to round up, life does nasty things. It does it mostly through complex 1 and complex 3 in terms of the oxidative stress caused by mitochondria. And there's good evidence that drives a whole bunch of nasty stuff. And therefore, there's good evidence that we should be able to ameliorate some of those problems by converting our compounds into drugs, the cycles and the sequels. So that's what I do nearly all the time at the moment, is collaborate with other people in a drug development program to try and make effective drugs to treat a whole range of aging-related diseases. And we hope that in a few years' time, we'll get into the clinic and we'll actually make a difference. And if true, that would be an example of starting with looking at the bioenergetics and looking at the properties of the electron transport chain and translating it right the way through into a drug. So I'm hoping to do that before one of these nasty things gets me and I have to shuffle this mortal coil and leave it to younger generation to finish up. Okay. Any questions?
Martin, I have one question. This is almost.
Hi, Amos.
Yeah, hi. Yeah, great talk. The bulk of your experiments, you measuring peroxide, right? I'm just wondering, what are we missing by not measuring superoxide? I mean, I know you went into the assumption that most of the superoxide is turning into peroxide by the action of sod, but then you also highlight how superoxide is different than peroxide in the extent of damage it can inflict. So I'm just wondering, the fact that, again, we're just measuring peroxide, H2O2 production, are we missing a big translational, I would say, aspect here?
Yes, for sure. So we know that superoxide in the matrix really is important. And the reason we know that is if you knock out the superoxide dismutase in the matrix specifically, then you get a mouse that can't last more than about 8 days, it dies. So having high levels of superoxide in the matrix that you can't clear is lethal.
Right.
And that's different from hydrogen peroxide. So in those mice, the hydrogen peroxide would either be the same or lower. So it's definitely a superoxide dependent effect. But we also know that the MCAT mouse, so a mouse where you express catalase specifically in the matrix, is protected against all sorts of pathologies, including various cardiac and metabolic pathologies. And under those conditions, it's the H2O2 in the matrix that's doing things, because the superoxide is not being affected by having more sod there present in the matrix. So we know that independently, both the superoxide and the H2O2 matter. So you're quite right that unless we measure the superoxide, we don't really know what's going on. We can only infer it. The big problem is, how do you measure it? And as you know, that's a difficult problem which requires probes which at the moment are not specific enough to be reliable. So we recognize that some people use probes that give signals that respond to superoxide changes, but we don't think that they're precise enough to use for anything where you really want to know what happened to the superoxide.
Right.
For example, some of the mitochondrially targeted superoxide probes, you can show in the absence of changes in superoxide, give signals in response to the redox state of the electron transport chain. So they're talking to something, cytochrome C maybe, and it's redox state. So the probes are reporting superoxide, but they're also reporting other things. And if you really care about what's going on, you may be misled by using the probes. So the answer to your question is yes, they matter. Yes, we would really like to know it. But no, we don't have a toolbox at the moment which allows us to do it unambiguously and conveniently. And I wish we did, because life would be a lot easier.
Right, but so just quickly a follow up on this. So you assume that the other sink to those superoxides are, R groups on amino acids, or there's anything else that, or lipids? I mean, it could be a whole slew of bioorganic molecules that can be that are used as a sink other than the H2O2, right?
Yeah. So what we know is that the mitochondrial superoxide dismutase is a very active enzyme and converts nearly all of the superoxide to H2O2, 99%. And we know also that even in the absence of that enzyme, the spontaneous dismutation of superoxide is relatively fast. So if you don't have SOD there, still most of it will end up as H2O2 just by spontaneous dismutation. But if you don't have the superoxide dismutase there, then the level of superoxide will rise. And as it rises, then all of the minor quantitative side reactions become quantitatively more important. And they're minor quantitatively, but they're hugely important pathologically, probably. So there's a whole range of that, and that chemistry has been relatively well investigated, because obviously the field of free radical biology is a huge field. So we know that superoxide is able to modify iron sulfur centers. So it knocks the iron sulfur center out of the aconitase, probably does the same thing for other iron sulfur center containing proteins, including complex 2, maybe complex 1. And it can also react weakly, but nonetheless importantly, with double bonds, particularly in lipids. So to trigger these lipid peroxidation cascades. And H2O2 is very good at doing that. And H2O2, if there's a bit of iron around, does Fenton chemistry, which is very damaging to a whole range of different molecules. So there's a lot of different processes going on, and it's not so clear which of them we really should be caring about. So that's to be established, but it's quite well known what those processes are.
Thank you.
Okay, so if there's no more questions, then I'll hand over to David, who's going to give his next lecture.
Okay, hello everybody. I'll just start my screen sharing as usual. Can everybody see my screen? Yes?
Okay.
Yep. Okay. So what we're going to do in this problem solving is I have not given you the full PowerPoint because the PowerPoint contains answers. Instead, I've given you a very short PowerPoint, which you should have available during the breakout sessions, which pose the specific problems that we're going to be discussing. And I hope that the Brand Lab will be contributing to the breakout sessions for expert advice for you students. So what we're going to do now is work with the seahorse respirometer, and we're going to put it to work to look at intact cells. Is everybody okay? Does anybody need a sort of comfort break for 5 minutes, by the way, or shall I start straight away? Everybody okay? Difficult to know the answer, isn't it? Yeah. I'll proceed. So this is the cellular complexity, which I've mentioned before in terms of the, in this case, a neuron with the proton circuit central and a vast variety of transport processes, either driven directly by the proton circuit or indirectly by ATP generated by the proton circuit. and the respiration of the cells can be quantified on the seahorse, and if we allow for non-mitochondrial oxygen uptake, this gives us the aggregate proton current of all the proton circuits in our incubation. And then tomorrow we're going to be dealing with the voltmeter in the similar system, measuring the plasma and mitochondrial membrane potentials. So the classic experiments with isolated mitochondria, I showed this yesterday, originated actually in 1955. What's that, 65 years ago, with the wonderful scientist Britton Chance, who did so much to establish the basic principles of electron transport. And this shows the classical experiment where the addition of succinate or substrate to mitochondria gives a slow rate of respiration, which is restricted by proton re-entry through a proton leak. There is no ADP present, so there's no possibility of generating ATP. The addition of ADP causes a state 4, state 3 transition, sometimes called a state 2, state 3 transition. One gets rapid respiration because now the ATP synthase is making ATP, protons are entering. This causes a slight drop in membrane potential, which returns when we run out of ADP. And then at the end, traditionally one adds a bucket of FCCP, which collapses the proton motive force, the membrane potential, respiration speeds up until you reach anoxia. And these are the classic experiments I would think that virtually all of us has done as an undergraduate in basic bioenergetics. The problem we are faced with is a problem of geometry. Isolated mitochondria, or some bulk cells, can be obtained in sufficient quantity that you can just put them into a chamber, close the chamber, and measure the oxygen uptake. The problem when one wants to move from isolated mitochondria in suspension to cultured cells on a monolayer is that we're moving from 3 dimensions to 2 dimensions, and the cells are available in a very limited yield. The classical experiment for isolated mitochondria, the classical oxygen electrode, again has this 3-dimensional chamber. I mentioned briefly the first approach that our lab made some 15 years ago to measure the respiration of cultured cells on monolayer by superfusing a very thin layer of medium slowly across and measuring the decrease in the downstream oxygen relative to the upstream oxygen. And this gives us a continuous readout of the respiratory rate. That, of course, has been supplemented or superseded by the seahorse, and the essential feature of the seahorse is sensitivity. That we have a monolayer of attached cells, or even mitochondria spun down, as Martin talked about yesterday, and the volume of this chamber is a matter of a few microliters. I think it's about 7 or 8 microliters for a standard 24 well. And we have the ability then, when the chamber is down, to very sensitively detect the fall in oxygen tension in that very limited chamber. And as Shona will be telling us later, one can also get a lot of information about the change in acidity, the change in external pH. And then we can raise the piston. When we've raised the piston, we can make an addition through the addition port, bring the piston down again, and then do the next measurement for the next few minutes. So this is really a wonderful system which works continuously, and I think quite a few of you will have the seahorse, and you know, if you have a few $100,000 to spare, then I strongly recommend it. That will skip actually. So when the seahorse was developed in about 2007, 2008, they came to us at the Buck and said, we've got this nice machine. We don't quite know what to do with it. We want to measure something like mitochondrial function. What do we do? And what we devised was something that we call the cell respiratory control experiment. And they decided to call their mitochondrial stress test, which I'm not quite sure how we're being stressed here. But the findings are very straightforward. And we start off with the basal respiration. And the basal respiration under most conditions in most cells is strongly controlled by proton re-entry, both through the ATP synthase and the endogenous proton leak. The addition of oligomycin removes the ATP turnover component and leaves us with the proton leak. And the difference is the amount devoted through the ATP synthase. This is approximate because, as I showed in the sort of previous slide a few minutes ago, the proton motive force, the membrane potential, changes on addition of oligomycin. There's a slight hyperpolarization. And the proton leak is highly dependent on the magnitude of the proton motive force. And so if you don't correct for that, you find that the proton leak you are measuring is at a slightly higher, driven by a slightly higher potential than the basic respiration. So it tends to overestimate the leak and underestimate the ATP turnover. And then the point I emphasized very much in the earlier lecture this morning is the spare respiratory capacity, the addition of FCCP to generate maximal respiration, and the difference being the spare respiratory capacity. Ideally, we would like to add ADP, but ADP does not get into the intact cells. And so our surrogate is to add FCCP and to measure the maximum respiratory capacity. And for most, but not all cells, that can be roughly translated into the ATP generating capacity. And then we terminate the experiment with a cocktail of electron transport chain inhibitors, and we're always left with some sort of basal respiration, which we always assume to be non-mitochondrial because we've added the inhibitors, and we subtract that from all the other rates. So this is every one of you who's working with a seahorse on intact cells will be familiar with this basic cell respiratory control experiment, which we introduced to the seahorse community about 12 years ago, and they've been using ever since. And what you get out of a single experiment are really important parameters, equivalent to those which Martin would have talked about with isolated mitochondria. We can estimate ATP turnover, proton leak, maximal respiratory capacity. You saw in my previous lecture this morning, using my old-fashioned technique, the addition of monochlorobimane, we can measure continuously the decline in ATP turnover when we remove glutathione in that particular experiment. So there's a lot of information available from this. As I said, in order to get the maximal respiration, we add FCCP. And the amount of FCCP that we need to add in a seahorse experiment for a cell is really, really critical. This is an actual experiment with cultured neurons. This is the estimated proton motive force in millivolts, and this is the respiratory rate. And as we start off in this situation showing high membrane potential and reasonable respiratory control, we so slowly titrate in FCCP, and we get to a threshold, which might not be quite as abrupt as it's shown in this slide. And any addition of FCCP above that threshold can cause no further increase in respiration, which is already maximal, but causes a dramatic drop in proton motive force. What we always want to aim in a seahorse experiment is to get precisely to that elbow where we have just the maximal respiration without any excess. If we have an excess, the proton motive force drops, particularly the pH gradient across the mitochondria tends to collapse or even reverse. And as substrates, most substrates such as pyruvate are driven into the mitochondrial matrix by the pH gradient, this means you immediately start to have bioenergetic effects because you're starving the cells, the mitochondria, of glucose-derived pyruvate. So one needs to take great effort to get just precisely to that threshold. The other thing which I actually tripped across, which surprised me, and I started developing a technique to measure both plasma and mitochondrial membrane potentials simultaneously with a combination of fluorescent cations and anions, is that you only have to got a slight excess of FCCP concentration above this threshold, and the plasma membrane potential collapses. Not only the mitochondrial membrane potential, but the plasma membrane potential. So a 10-fold excess of FCCP above the minimum amounts to remove respiratory control actually collapses the plasma membrane potential. And if you're working with a cell and you want the effects to be restricted to the mitochondria, that's going to have dramatic effects on the cell function, on the respiration, on substrate transport across the plasma membrane, etc. So be very, very careful with your FCCP additions. The other point, which I'm sure you're aware, is that if you're working with a nice mitochondrial inhibitor, antimycin A, rotenone, oligomycin, then under most circumstances, those are highly selective for the mitochondrial inner membrane. In other words, you can add rotenone, and you can be sure that the only thing that's going to happen is that you're going to inhibit complex 1. Now, the problem is that ionophores show little or no membrane selectivity. And so what that means is if you add FCCP to a cell, however careful, I've already said it gets into the plasma membrane. It will also get it, for example, if you're working with a neuron, it will get into the synaptic vesicles and release neurotransmitter by collapsing the proton gradient across the vesicles membrane. Valinomycin, which is a potassium ionophore, which increases the potassium activity of the inner membrane, you can use it with isolated mitochondria in a low potassium medium. If you add valinomycin to an intact cell, because the mitochondria are sitting in a high potassium cytosol, the mitochondria will collapse their membrane potential and actually swell and explode in the presence of valinomycin. Valinomycin will also work at the plasma membrane, but it's a bit useless if it's already killed the mitochondria. Ionomycin is interesting. This is a calcium to proton exchange ionophore. And the last time I looked at this, which was of course last year, it had something like 22,000 citations, 22,000 papers have utilized ionomycin and used it almost exclusively to increase cytosolic calcium, because it exchanges calcium for 2 protons and does increase the cytosolic calcium. Because it shows no membrane selectivity, it also gets into the inner mitochondrial membrane, and there it sets up a dissipative cycle between the calcium uptake pathway, the calcium uniport, and ionomycin itself. So it acts as a very potent calcium-dependent uncoupler of mitochondria. And it's interesting, out of the 22,000 papers on ionomycin, there are 100 that actually mention this possibility of ionomycin uncoupling, and we were responsible for a handful of those. So ionophores can seriously damage your cell's health. You have to use them extremely careful, including FCCP. This is the first sort of warning point you're going to come across in terms of seahorsery. Now, I've spent a lot of time working on pancreatic beta cells and beta cell lines. And one of the characteristics of insulin secreting cells, they have very low lactic dehydrogenase activities. They want the rate of glycolysis to be directly transported, if you like, into the mitochondria to control mitochondrial ATP production. They don't want any sidelines of anaerobic glycolysis. And one of the things one has to remember when you do the classic respiratory control experiment with a cell is what happens in that time after you add the oligomycin. After you add the oligomycin, you totally block oxidative phosphorylation. And in the average cell, all the basal housekeeping ATP requirements of the cell has to be generated by anaerobic glycolysis. And we know the rough figures: 2 ATPs per glucose from anaerobic glycolysis and 30 odd by oxidative phosphorylation. So in theory, glycolysis has to accelerate 10 or 15-fold after the addition of oligomycin to maintain the ATP levels. And this is something which Shona will be dealing with in great detail shortly. If you don't have lactic dehydrogenase, that doesn't work. You can't complement the lack of oxidative phosphorylation by increased glycolysis. And so you get an ATP crisis. An ATP crisis means ATP levels fall. Because you need ATP at 2 stages of glycolysis, hexokinase and phosphofructokinase, then glycolysis fails, and you find the rates that you get, the FCCP rates you get after a period of oligomycin, are greatly depressed. What you do to overcome this is you add oligomycin and FCCP simultaneously in one set of wells and just oligomycin in another set of wells. And the hope is the simultaneous addition means that you can generate the maximal respiratory rate before there's time for the ATP levels to actually collapse. So you've got to modify the cell respiratory control experiment for certain cells. Very careful with the FCCP and careful with ATP depletion following the addition of oligomycin. Now, what we're going to do now for the next few slides is to take a case study, because it's something which I worked on. So 2008, 2009, the only experiments which were done in the seahorse were cultured cells which were grown on the cover slips. What we discovered was that you could take free floating isolated nerve terminals, so-called synaptosomes, isolated nerve terminals, which I've done a lot of work on, centrifuge them down onto seahorse plates, and they remained attached, and you can actually measure their respiration in the same way that you could measure with a cultured neuron. And this, of course, we extended this to mitochondria, and now a high proportion of the work that's done with seahorsery is done with isolated mitochondria spun down onto the seahorse plate, as Martin has already told you about. So one can take a synaptosomal pellet and you spin it. And interestingly, if you want to measure volts and amps, as we always do, if you spin the pellet in an ionic medium, you get aggregates of synaptosomes which adhere to the bottom of the plate. And this is very good. This gives a high yield for measuring in the seahorse. At the same time, you can spin in a non-ionic medium in sucrose in a multi-well plate, and then you get single discrete synaptosomes which you can use to monitor membrane potential, calcium, etc. I'll give you an example of this in a bit. So it's quite a versatile technique, but we're going to focus largely on the seahorse. So even if you are in suspension, many cells, mitochondria, synaptosomes can be attached to seahorse plates by careful centrifugation. Now, 50G, 100G, you don't have to go crazy for most cells. So what we're going to do now for the rest of this practical is we're going to show you the results either of actual experiments or simulations. And we're going to start off with things which are relatively simple, and then they're going to get more complicated. And what you're going to do, you should now make sure that you've downloaded from the Dropbox the student problems for the P2 practical. And then when you disappear into your separate breakouts, you're going to have a period of time, 5 minutes, to discuss as a group. And I hope Martin's students will be looking in as well to advise and help and confuse. And then after the 5 minutes, we're going to end the breakout session. We're going to come back to the main session, and we're going to discuss the results and discuss your conclusions and see whether you were right. We're not looking for precise compounds. There's no way other than luck you'll come across precise compounds, but it's compounds that act sort of as though they were. So they act sort of as though they were rotenone or as though they were antimycin A. So you won't get the exact compound unless purely by chance, but we want you to investigate and discuss what you think, what type of compound is being added. And this is the reference slide, if you like, which is going to appear in a lot of these, slightly idealized. It's the basic basal respiration, the addition of oligomycin, the addition of FCCP, the addition of rotenone antimycin A to get back to the basal respiration. And we're going to start with a pair of problems. The first problem is what's happening here. Okay. The second problem is what's happening here. And here we're showing the oxygen consumption rate and the ECAR, the extracellular acidification rate. And the control is in red and the experimental is in blue. And now if everything works, then Ricky is going to put you out for 5 minutes into your breakout sessions for you to discuss problems 1A and 1B on the PowerPoint that you can download from the Dropbox. So Vicky, can you start that now, please? Happening with problem 1A. Anybody from Group H, please? Anybody from Group 8?
If you're not sure, Group 8 is Ritesh, PK, Manish, Amanda Lynn, Robin. You guys are Group 8.
Okay, waiting.
Actually,
yeah.
Not sure about the first profile, but looks like it can be some substrate which has been added over so that there's a slight increase in the oxygen consumption rate.
Okay.
Or even we were thinking like can be like ADP or something which has been added.
Well, these are intact cells, so the ADP won't get in.
Yeah,
that way.
Yeah, for the first one, we could just assume that there is some substrate which has been fed in at that port.
You're right. So let's go straight to the answer. And what it was adding, on top of glucose as initial substrate, adding pyruvate in addition. And pyruvate is giving an increase in initial respiration. It's giving a very profound increase in maximal respiration. It's providing lots of extra electrons there. But the puzzling thing is that it's increasing the oligomycin insensitive respiration. And what does that mean? I mean, does it mean that pyruvate is a weak protonophore if it's acting like a very poor version of FCCP? Okay. So in other words, what do you think is happening here? But let's put this out generally. I think it's going to we're going to be a bit too claustrophobic if we ask specific groups. We ask generally the plenary session to come up with suggestions here. So we move on. The addition of pyruvate has roughly doubled the proton leak across the inner membrane. Any ideas what's happening?
Maybe they were more polarized and there was a greater amount of protons to leak when oligomycin was added.
Okay, you win the jackpot. Basically, you're absolutely right. I put this one up because I want to emphasize the importance of thinking about membrane potential, proton motive force, thinking about the voltmeter as well as the ammeter. And what happens is that pyruvate is a very potent substrate, and together with glucose, the mitochondria maintain a higher proton motive force, and the proton leak is very dependent on the proton motive force. And we can see this with some experiments which my colleague Akos did in this project. This is the individual synaptosomes spun down on a plate I told you about and measured with TMRM as a membrane potential indicator, and just in this case qualitatively, but demonstrating that addition of pyruvate hyperpolarizes the mitochondria within the synaptosomes. So they maybe have another 5 millivolts of proton motive force, and the leak is very voltage dependent. So that's why you get a higher leak. So the ammeter is great. Whenever possible, do voltmeter experiments as well. Otherwise you can get yourself really confused. Okay, just briefly to sort of sidestep here, you very often see traces normalized to 100%, oxygen consumption rate as a percentage. And this is an experiment where from the beginning, we had glucose in the blue trace, we had pyruvate in the red trace, and it shows again the dramatic effects of pyruvate on maximal respiration. Is it valid to normalize those two? Is this a valid approach on the right? Somebody say yes or no and explain. In other words, are you allowed to normalize?
Dr. Nichols, could you please share your screen?
Can everybody? Can nobody see my screen?
We can.
Oh, right. I'm sorry. This is. Can everybody see my screen now?
Yes.
Okay, I apologize for that. You said that earlier. I should have actually noted it. I'm very much a beginner with Zoom. So, okay, the two traces here, which are glucose as initial substrate, pyruvate as initial substrate. Is it valid to normalize these two in this particular experiment? A yes or a no will suffice.
I think it's, as a toxicologist, I get this question a lot because it depends on what you're looking at. If you're looking at the effect of an acute injections, you want to establish the baseline beforehand, it could work. But here you've got differences even at a baseline respiration. And so you're going to miss out on those differences if you do this 100% graph.
Yeah, you're right. It's totally invalid. X. You can only normalize traces if their initial conditions are identical. So if you started off with just glucose, and at this point you added pyruvate, for example, then because they're starting at the same level, they're both starting identically, then you can normalize. This is totally wrong to normalize under this condition. Okay. So you can only normalize two seahorse experiments if the starting conditions, cells, media, and additions are identical. So let's move on to problem 1b. My apologies for forgetting about sharing screens, but now I'm sharing the screen, I hope for everybody. And again, let's have a sort of open session for anybody to discuss what they think the addition, the question marked addition was at port A in these two experiments.
Some sort of uncoupler.
Yeah, well, so you think it's uncoupler. So explain why you think it was uncoupler.
I'm just saying this based upon the OCAR profile, oxygen consumption rate, that when you add, the oxygen consumption rate is shooting extremely high.
Yep, so respiration shoots up. What happens to the acidification rate? And we're assuming as the approximation at the moment that the acidification rate is a measure of glycolysis. Now, Shona will tell you in a couple of hours' time that that is very approximately incorrect. But at the moment, we'll take that, that assumption that this represents anaerobic glycolysis lactate production. So why is that shot up with the uncoupler?
I'm not sure about that.
ATP is not being generated due to the uncoupler, dispensing the proton motive force.
Yeah, so it's the Pasteur effect, if you like. Glycolysis accelerates to compensate for the lack of oxidative phosphorylation. Now, I think we'll leave it at that. So basically, this is the effect of a protonophore, big increase in respiration you would expect, and at the same time a big increase in acidification, largely but not entirely due to the production of lactate in anaerobic glycolysis. And so it should be no surprise to anybody that that was FCCP in that particular case.
So is there a reason that upon addition of FCCP at C, it seems like the oxygen consumption trace is going lower and lower?
Which is here, you mean?
Right, yeah, yeah.
There is, let me think this through, basically. The most dramatic effect is with the second edition of FCCP, the normal edition of FCCP, and that's where you get the big drop in respiration. This is a bit what I was talking to you about, that if you have an excessive FCCP, the proton motive force collapses, the pH gradient reverses, and what is driving this respiration, even though the substrates glucose and pyruvate, the pyruvate is now having difficulty getting into the mitochondrion because the pH gradient is actually reversing, and a normal pH gradient drives the uptake of pyruvate into the mitochondria. And so what part of this effect is due to the difficulty of pyruvate getting into the mitochondrial matrix. Okay. So let's, this is a bit clumpy, but we're going to improve as we go on. And like that, we'll forget. And we're going to go on now to the next pair, which are going to be problems 2A and 2B. These are going to be, again, fairly straightforward. So you're good about the first one's an additional substrate for 1A, and additional protonophore for 1B, and you see the sort of characteristic responses. And the next session, which again we'll give 8 minutes for, are problems 2A and 2B, which you should have in your PowerPoint. Here's the addition of 2A, and we have to guess, or again, the red trace is the control, magenta trace is the experiment. So in the magenta trace, something was added at A, and we're seeing the effects on the oxygen consumption rate and the effects on the extracellular acidification rate, on the assumption that that is a rough measure of lactate production that will be refined by Shona. So let's now, that was 2A. This is 2B. In this case, the unknown is in sort of rather pale orange. And again, we're looking at the oxygen consumption rate and the extracellular acidification rate. So if you, Ricky, if you devise the, switch us into the breakout.
Wondering, could you please share the glutamate concentration reference with me? Because I was looking for it and I might have missed. And
so Which one was this, the glutamate?
And so you had the synaptosome figure where you showed the distribution of overall glutamate in the vesicles, synaptic cleft, and cytoplasm in like millimolar, micromolar. That would be really great.
Yep, that wasn't an. You wanted the slide, or what do you want? Because
the citation.
No, that was purely out of my head. That was.
Oh, I see.
Yeah, no,
okay. Yeah, the reason I find that very interesting, we just started doing metabolite tracing with one of the pathways that we're studying with cultured neurons.
Yeah.
And it seems like it is in response to glucose uptake, it's creating more glutamate synthesis, and we think it's kind of regulated through the glycolytic compartmentalization and hexokinase activity. And I'm just at the beginning of learning more about glutamate concentration and synthesis.
Okay.
So we think this pathway may be in neurons creating signal. Okay, there's excess glucose, can be utilized for anabolic processes, just stock up glutamate. But my knowledge is very limited. We just stumbled upon this, and
we have an awful lot of glutamate in
I know. Yeah, so I think it will be interesting to really figure out what is the defining point and how neurons can decide whether it's time to create more or store more, like what is regulating the flux in a way, starting from glucose. That's where we're going, at least.
Okay,
because
that's interesting. Yeah. Here comes everybody back. Okay, welcome back, everybody. We're slowly getting a little more fluent in this. So I will remember to share my screen, as I forgot beforehand. There we are. I hope everybody has the screen. And welcome back, it says. And again, this will work more fluently if we have a sort of plenary session for some spokesman from one of the breakout groups to talk about problem 2A. What's happening in problem 2A? Okay. Anybody?
So so we think that 2A.
Brandon, hi. Yeah.
Yeah, we think we're looking at a ATP synthase inhibitor because we see a decrease in the oxygen consumption rate like we do with the oligomycin addition, but it is still sensitive to uncoupling, so we can still get maximum respiration. And kind of the same thing with the extracellular acidification if we assume that we're looking at glycolytic acidification. The same effects as oligomycin are happening.
Okay, so it looks as an ATP synthase inhibitor. Is that a sort of general consensus in the group? It's difficult to know unless everybody shouts yes.
I guess one question.
Yeah.
One question our group had was, I guess the thought was also, say if it was a complex 1 inhibitor, would the cells be able to completely bypass it with complex 2 to get the same maximal response?
These are intact cells. Okay, you did isolated mitochondrial experiments earlier with Martin, where you can feed succinate in to bypass a complex 1 block. With the proviso that Martin was talking about, these very interesting effects of ischemia reperfusion and a sort of buildup of succinate in the general cell. Under general conditions, the only succinate you get is from NAD-linked reactions in the citric acid cycle. So the moment you add a complex 1 inhibitor, then you block the citric acid cycle and you block the production of succinate, so it can't actually compensate.
Got it.
Yeah, so the answer, it's an ATP synthase inhibitor. And of course, what we simply did was to give you additional oligomycin followed by oligomycin. Again, it's straightforward, but it's fairly informative. You get virtually no effect on the maximal respiration. So you basically just double the oligomycin concentration there. And the effects we'll just focus on this basic effect on the ECAR rate this is going to get us into Shona territory, so I'll leave her to discuss these sort of effects after lunch. So basically, you inhibit oxidase phosphorylation, glycolysis enhances to try and compensate in these particular cells. So that's 2A. Let's talk about 2B here. Again, anybody, a spokesperson for any of the other breakout groups can discuss what's happening with problem 2B.
So our breakout group thought this was, this is Gavin, that this was either a complex 3 or complex 4 inhibitor. And we were uncertain that we'd be able to distinguish between the two with this experimental design.
Okay, so it's an electron transport chain inhibitor, complex 3, complex 4. Could it be a complex 1 inhibitor as well, or just 3 or 4?
We thought 3 or 4 because with pyruvate in the substrate mix, we would see some succinate being formed through the citric acid cycle.
Yeah, this is the same point that we were discussing earlier, that the only succinate you get is due to the operation of the citric acid cycle, which involves NAD-linked dehydrogenases. So a complex 1 inhibitor would be immediately stop any succinate formation. You wouldn't from pyruvate. So, but I'm very pleased that you are not being rigid. You're thinking about multiple possibilities here, because it is clearly an electron transport chain inhibitor, and it was the old-fashioned complex 1 rotenone, but you get equal results if you inhibited complex 3, mixothiazole or antimycin A. You can inhibit complex 4 with cyanide or azide, and you essentially get the same sort of results, except cyanide and azide will probably screw up the acidification trace. So yeah, it's an electron transport chain inhibitor. And it's interesting, you get this very stable baseline. I think these were C2, C12 cells here, which you, Martin mentioned earlier this morning. And this is the non-mitochondrial respiration, which is sort of essentially constant throughout. Okay, folks, those were the easy pair. Now we're going to give you one at a time. We have, I think, 3 left to go. And Ricky, how is our time going? Difficult to translate here with Central European time. Ricky? Is Ricky there?
You started late, so if we were to keep on schedule, you'd have 15 more minutes, but you started your session a little bit late.
Right, I started about 30 minutes late, didn't I?
Okay, so then if you add on 30 minutes, you've got about 45 minutes, but people
okay,
might
so I will be about 20 more minutes, I will guess, to do these last 3 problems.
Sorry, just on the schedule really briefly, the next thing is lunch. So what we could do is shorten lunch a little bit and then start the after lunch lecture on the same time schedule, just to try to keep
Okay, so how much time does that give me, Shona?
Take the time you need, David. So about 20 minutes is fine.
Okay, right. Well, we better get started then. Here's problem 3. And again, red is basal, and magenta, whatever you call it, so purple is the experiment. And this demands quite a lot of thought. So we'll give you the, let me think, 3, 8. Let's make it 6 minutes, because then you won't miss your lunch. Six minutes for this one, Ricky, if you can organize that.
Yes.
Let's break out into a session for 6 minutes.
Okay.
See my screen?
We can.
So, yeah. So again, let's just have an open session, preferably somebody who hasn't come up with an answer yes. What is the curious compound we're adding on the first port here? what type, what is happening?
We'll take a shot in the dark here, and we'll say a PDH inhibitor.
Okay, so a pyruvate dehydrogenase inhibitor. So that will, the idea is that limits the electron flow through the electron transport chain, so you get a decreased respiration. How would glycolysis react to that?
Our guess was that if you had an increase of pyruvate in the cells, maybe it's not glycolysis we're seeing altered here, maybe it's the lactic acid export.
But pyruvate would not infect glycolysis, right? We were thinking it'd be some inhibitor of substrate upstream. It has something that reduces both respiration and reduces glycolysis. And we came up with something like inhibitor, glucose uptake inhibitor, or maybe 2-deoxyglucose.
Yeah, no, it's an interesting idea. Maybe one needs to go downstream, basically, and think about the fact that if we compare these two traces, what does that tell us about ATP turnover in these two conditions?
It's lower in the treatment.
So the treatment has lowered ATP turnover. If it's done it by screwing up the bioenergetics, you would expect glycolysis to increase as a compensation. Now, it's a bit noisy trace, but actually glycolysis decreases as well.
It can't be an electron transport chain inhibitor or an uncoupler.
I don't think so either, no. This could be an interesting clue, that when you add oligomycin, you still have a lower rate of, let's call it glycolysis than you do in the control. So glycolysis is depressed in the presence of this compound. Respiration is depressed in this presence of this compound. ATP turnover is depressed in the presence of this compound.
Could it be a hexokinase inhibitor?
I don't think so. I have to think about this. But why do the cells produce ATP? Silly question.
To do work.
To do work. These are cell lines which grow.
I see. ATP demand is lowered, you're saying.
Yeah, ATP, that's just the clue. The ATP demand is decreased. And it's decreased because we've done something which has decreased the ATP demand.
So inhibition of translation or rapamycin, something like that?
Yep, something exactly like that. And if
I
we
was
look...
guessing something like cyclohexamide or...
Right, translation.
Oh, how clever. Who's the clever guy who said that?
Dad, you can't give away the answers.
Oh, sorry. I was...
Yeah.
I have a question.
Yeah, it was a protein synthesis inhibitor, and these are actively proliferating cells. Are putting a lot of energy into producing new material. So a lot of the ATP is going towards cell proliferation. Somebody had a question?
Is there any reason this could not be a plasma membrane, an inhibitor of plasma membrane pyruvate transport?
Well, you've got glucose as well, but so if the pyruvate, trying to think back to the experiment I showed you at the beginning, the very first one.
Well, pyruvate increased OCR in addition to glucose, and so withdrawing that could decrease it. Oh, but actually, oligomycin was different in that experiment.
Yeah, and this is not significant, this difference in maximal respiration.
Yeah.
The bars overlap, so you're not getting a sort of effect of lack of pyruvate, which you would have, which would mean that the magenta trace would be really depressed in the presence of FCCP.
Well, does that mean when you inhibit protein synthesis, why would you not reduce that much more? I mean, going back to your point that the bars are overlapping, wouldn't you? I mean, how much?
Because the mitochondria are not being affected. So you, in the presence of FCCP, you're simply saying, you know, what's the maximal rate of respiration? So the mitochondria is an acute experiment. I mean, the mitochondria haven't had time to change.
Neither,
So
and there's plenty of substrate, you're saying, which rules out that it's a big effect on the substrate.
yeah, that's right.
But I'll
Anybody-
just add, sorry, really quickly, that this is a really good example of, this is real data, or these are real data, they're messy. I mean, remember that we're asking you, if you look at the right-hand trace, to identify a meaningful difference after oligomycin is added, but in theory, there's no meaningful difference after FCCP is added. It's hard to see that because it looks like there's a pretty big difference in both cases. But in theory, after you add FCCP and the rate of respiration is no longer controlled by ATP demand, these rates should be the same. And same for the uncoupled rates in the respiratory trace. Those rates should be the same. If our theoretical model is correct that that cyclohexamide is the reason for depressed rates of both respiration and glycolysis after it's added. And in reality, it's likely that the lack of protein synthesis by the time you get to 40 minutes or an hour after its addition could be having effects on these traces. We just don't know, but it illustrates the idea that the further out you get from adding something that's really bad for the cells, the less reliable your traces then start to become.
Okay, fine. I'm a bit worried about your virtual lunch. Let's group together the last two problems. I was going to have those separately, but let's have, Ricky, let's have 8 minutes. Oh, sorry, first of all, before we do that, how do you distinguish cyclohexamide from low oligomycin? This is oligomycin, and it shoots up the lactate production. And this is cyclohexamide, and it decreases the lactate production. So it can't be oligomycin simply because the ECARs go in opposite directions. I just wanted to mention that in passing. So Ricky, now let's move to the last two. Let's group the last two together, and we'll have, oh, can we have 8 minutes, or do you lose lunch after 8 minutes?
No, there's an hour set aside for lunch, so 8 minutes is fine.
Okay, let's have 8 minutes and tackle problems 4 and 5, the last two problems, tackle those at the same time. And these, again, it's real data. This is actually my synaptosome data. So we're dealing with a sort of neuronal system and all the complexities that has. You might sort of think a little bit about some of the things I talked about in my first lecture today on neuronal bioenergetics, but see how you go with these two. These are really probably most important two, these two. 8 minutes from now.
So no breakout rooms.
Okay, every time I do it. Okay, got it? Right, good. Right, let's have somebody to talk about problem 4, please. There is a logical progression of questions to ask.
Is it more than one thing you're adding?
No, it's a single clean compound. Now you ask the questions: what happens to the basal respiration? What happens with oligomycin? What happens with FCCP? And work your way through sort of logically through these. And these are just two different concentrations of the same compound at 1 micromolar or 5 micromolar, just the way we happen to do the experiment. Okay, well, let's answer the first question. What's happening to basal respiration? Okay,
there is more energy demand, so there is increase.
More energy demand. So, yes, explain what you mean by more energy demand.
So, I think there is, after addition of this compound, there is ATP crisis or something, so it increases the respiration.
So the respiration is increasing. Now,
yeah.
Respiration can increase for one of two reasons. If we forget about this hyperpolarization business I talked about earlier, it can increase either because ATP turnover increases or because proton leak increases. So what's happening in this particular experiment? Which?
Sorry.
So anybody?
ATP, you actually have an increase in ATP-linked respiration.
A dramatic increase. It goes up about 3 or 4-fold, doesn't it? There's the control. And there's in the present these two concentrations. So something has increased the ATP demand 3 or 4-fold in this particular experiment. The clue I gave you that these are synaptosomes, these are isolated nerve terminals, and we're dealing with something rather like I was telling you with glutamatic cytotoxicity, except this isn't glutamate or anything like that. So we're adding something which is greatly increasing ATP demand, and there's no sort of inherent reason why you would know the answer.
Is it sodium?
Sorry?
Is it sodium?
It's something to do with sodium. And yeah, I mean, I'll tell you the answer because it doesn't sort of detract from the other questions I'm going to ask. This was, come back. This was an alkaloid called veratridine, which opens up, activates the sodium permeability or the activity of the sodium channel in the plasma membrane. And then you get this dissipating ATP-driven sodium cycling through the sodium pump and back through the sodium channel, using up a lot of ATP. So it's rather similar to what was happening with the NMDA receptor, but there's no reason why you would know that. But the key point about this is what's happening with FCCP. Why is the FCCP rate depressed in this experiment in the presence of veratridine? Again, it's something we've touched upon earlier in this problem solving.
Maybe it is depolarizing the mitochondrial membrane, similar to what FCCP does when.
Well, FCCP will depolarize the membrane, but what we're observing is a decrease in spare respiratory capacity. So in other words, these synaptosomes, their maximal respiration is less than those synaptosomes. And anybody got any idea of what is happening? We talked about it a bit in relation to beta cells. And I think Shona also mentioned this.
Could be toxicity.
Yes, well, it's a sort of toxicity. It's not. It's creating a stress on the nerve terminals.
They don't recover as the recovery of the nerves.
Yeah.
In a sense, that's the stress.
Yeah. Let's talk about ATP. So here we have a given rate of ATP turnover, and we've established that the ATP turnover goes up about 3-fold in the presence of veratridine. And oligomycin doesn't stop veratridine working, but it switches the whole bioenergetics onto glycolysis. And these nerve terminals have quite active glycolysis. We said in the control case, switching from oxidative phosphorylation to glycolysis means that glycolysis has to accelerate about 10 or 15-fold. What's happening here in this experiment in the presence of veratridine?
Higher uncoupling.
Well, not so much higher uncoupling.
Translocator inhibitor, ADP translocator inhibitor in the cytoplasmic site.
Yeah, and all interesting ideas. Think about the ATP. Where does the ATP come from in the presence of oligomycin?
Glycolysis.
Yeah. And in the control conditions, with that ATP demand, glycolysis has to increase 15-fold. How far does glycolysis have to increase here to keep the cell, the synaptosome running with this wide open veratridine sodium cycling?
3 to 4 times the control?
Yeah, 40, 50 times. It's impossible. so that by the time you get around to adding FCCP, you have an ATP crisis, which means that your respiration rate is failing, and it's failing because there's not enough ATP to drive glycolysis. Okay, so this is, you would see this in any situation where the stress on ATP production is so severe in the presence of oligomycin that the cells or the preparation starts to fail at the time that FCCP is added. So the ATP is so depleted at this stage that glycolysis can't work. So I guess pyruvate is working, but the glycolysis is not. Is that okay? The rate is depressed because you put an impossible ATP demand in the presence of oligomycin by your design of the experiment. And as I said earlier, what you should do in this case is to have oligomycin FCCP added simultaneously, and you get probably the same maximum, and added sequentially. So you do 2 separate experiments. Is everybody okay with that? It's just an example of don't ask your cells to do impossible things.
I have a question. So with this FCCP treatment, don't you kind of unlink ATP demand from respiration, or do you?
Well, you let's think about that wording. Yes. I mean, there is still the ATP demand, and so this nerve terminal, the ATP demand, is to run the normal housekeeping and to keep this very energetically dissipating sodium cycling going at the same time. And then as soon as you add oligomycin, you have switched everything to glycolysis. The mitochondrial respiration is okay. This is a very low rate of respiration anyway, and that there's still enough pyruvate being delivered to the mitochondrion from glycolysis to drive this low rate. But when you ask for the maximum rate is severely depressed because that ATP level in the cytosol is now not sufficient for maximal glycolysis. That's inhibited. Are we okay?
So if maximum glycolysis, or I guess if our ATP demand has exceeded maximum glycolysis, why does that matter in the context of FCCP?
Because FCCP is telling you the activity of the electron transport chain, and together with the upstream substrate supply. And we are damaging the upstream substrate supply by depleting the ATP such that where glycolysis is failing. So we're getting rid of at least the glycolytic component of the substrate, that we're adding glucose and pyruvate, and certainly the glycolysis is going to fail. And if I had shown the ECAR acidification rates, you would have actually seen the same. You would have seen that in the presence of ratridine, the glycolytic rate that you get at this stage is severely depressed. Okay. Shall we proceed to the final one?
What will be the status of cell health here? Because we are activating the Na+ cycling, so it keeps going.
Well, they're dying rapidly at this stage. Yeah.
Okay. Yeah. I thought maybe because of this Na+ cycling keep going, the cell will die, right?
Yeah. You're putting an impossible strain on it. Okay. Let's move to the last one. This is equally important. And let's have some ideas of what sort of thing is in the question box here. And again, you have to be terribly generic. You can just say something is happening somewhere.
Something that's increasing ATP consumption?
If it was increasing ATP consumption, wouldn't you get an increased oligomycin sensitive respiration here? It looks so ATP turnover is just the same under these conditions.
Something that's dissipating ATP reserve based on the FCCP without increasing.
Right. So what should we be seeing? We're seeing the compound itself, when we add it, does absolutely nothing. You can't see any effect of that. You can't see any effect when you add oligomycin. There's no effect on proton leak. And yet, when you add enough FCCP, and this is enough to give maximal respiration, it's not that the FCCP is not working, the rate is severely depressed.
It seems like there's less substrate there that's able to meet that sort of maximal FCCP.
Yep, keep going.
Since the substrate is going to be either glucose that turns into pyruvate or pyruvate itself, it seems like we're gradually inhibiting possibly pyruvate import into mitochondria or some reaction that generates NADH is not able to generate NADH as well.
You're getting there. This tells us that we're doing something upstream to limit substrate delivery to the mitochondrion, or it could be the electron transport chain itself. Why do we only see the effect when we add FCCP? Why do we not see the effect at any of the other steps?
Because we've only partially inhibited this phenomenon.
Yep. And
could oxygen be the rate limiting factor here?
Not in this particular case. No. We're not sort of going anoxic or anything like that, or inhibiting cytochrome oxidase or anything similar. What it is, let's come back, is an inhibitor of a pyruvate transporter, which I think somebody came up with. It's called cyanosinamate. But the important thing about this is when you're designing an experiment, let's say you've got a nice job with a big pharma, and they're developing their drugs for billions of dollars, and you're testing them for primary toxicity on mitochondrial cell preparations. And you add the cyanosinamate and you send to your boss report saying it's perfectly safe, it doesn't do anything to basal respiration, doesn't do anything to oligomycin ATP turnover, go ahead and give it to the president, for example. And the reason is you only see an effect when you relieve the rate restrictions and the other steps so that all the flux control is at the point you're interested in. I tried to sort of emphasize this back at the beginning of the lecture, that you design the experiment such that the rate control is where you're interested in. Here, the rate control is proton reentry. Here, it's still proton reentry. And those rates are low enough that you don't see the effect of cyanosinamate. The moment you add FCCP, the rate control shifts from downstream, proton reentry, to upstream generation. And then you suddenly see this dramatic effect of cyanosinamate in inhibiting the maximal respiration. So it's really important for the design of experiments that you actually only, if you're interested in a particular step, you design the experiment so that that particular step is rate controlling. And in the case of anything upstream of the proton circuit, you remove the rate control by adding FCCP down at the bottom. Is that okay? Okay, so I have reached the end of the show and essentially the end of my vocalization, and I'm going to go to bed. But if anybody has any questions, we have a lot of time tomorrow for general debate. So I have to end now, and I will go to bed, and you will get on with your lunch, depending on where you are, unless you're living in Hawaii or Australia. And I will see you tomorrow morning, Pacific time, so I shall leave you now.
David.
Right?
Thank you. Everybody else, why don't we plan on reconvening at 2 o'clock? We'll do a nice soft start, so people who are coming back, take your time. I did want to talk a little bit about the structure of the afternoon. So yeah, I'll spend an hour kind of going over the theory of ATP production, how we measured in the seahorse, and how we associated with the rates that can be measured on a seahorse. Then we'll take a half-hour break, and then at 3:30 we'll start on the practical session. I do really want to stay on time, so I will do my best to do that. And like before, during the practical session, we'll have Brand Lab members seating the rooms, helping to prompt discussion, helping to head off unproductive hypotheses and ways of thinking about the questions. But as before, just as a quick reminder, please feel free to jump in and interrupt. Please feel free to ask questions during the time that you have them. It can be hard to get them to later. The context kind of loses a little bit. So yeah, please feel free to interrupt. Let's try to keep this as informal as possible. Just to let you know about my setup, I have 2 monitors. So when it feels like I'm looking away from you, I'm actually looking at the gallery view of all the participants. So, so if you ask a question and I look away from the screen, I'm actually looking at you while you're asking it. Okay, so the things to carry forward from the prior day and a half is really everything, but in particular, the discussions that we have had up until now of rates, of steady states, and of forces and fluxes. These are all ideas that are fundamental to how ATP production works in a cell and how we calculate ATP production from the rates that we can measure on the seahorse instrument. But I would like to say at the outset to keep in mind that even though this is going to seem a little bit like a seahorse lecture, it isn't. It's really a lecture about how rates in cells that are associated with ATP production can be used to calculate ATP production. So it doesn't really matter, and it shouldn't really matter, what instrument is used to gather those rates. As long as they can be measured simultaneously, the calculation and the theory behind it should still apply. So again, make sure, so I mean, many of you are seahorse users, but for those of you that aren't, don't feel left out. Any instrument, again, that can be configured to measure oxygen consumption and proton production in some sort of medium could in theory be used to calculate ATP production rates, as long as they can be calibrated to absolute units. So with that in mind, let us proceed. Okay, so steady states are often difficult to think about, but there's a reason that this is funny. And the reason that this is funny is that you already understand steady states. So if you do, great. And if you don't think you do, you actually do. The reason that jokes are funny is because they violate an expectation. And your expectation here is that there is a steady state, and that steady state is 0. So the steady state accumulation of candy by Lucille Ball should be 0, and it's not, and that is why it's funny. And so to the extent that a rate in contributes to an intermediate level of something that's being dissipated by a rate out is something that you inherently understand. Even before we took this course, even before you took biochemistry, this hopefully would have been funny. So again, the rate in, the influx, to produce a level or an intermediate that then gets dissipated by efflux is the same principle that applies to proton motive force accumulation and dissipation and ATP production and consumption. Okay, so similar to the modular kinetic analysis and the modules that Martin talked about yesterday, we can think of ATP production similarly, again, in terms of modules. So instead of substrate oxidation, leading to a common intermediate of PMF dissipated by two different branched pathways of consumption, we sort of have inverted this process. We have two major pathways of ATP production. J, again, refers to rates. And we have glycolytic ATP production and oxidative ATP production. Together, these two modules contribute to the production of ATP. So ATP levels are not a great measurement because ATP, the nucleotide levels remain relatively clamped in the cell. It's more helpful to try to measure something like an ATP-ATP ratio. We talked about ways this is easy and ways this is not easy yesterday a little bit. But this is what the cell cares about. The cell cares about how much ATP there is relative to how much ADP there is. And then of course, ATP is consumed by all the processes that use ATP to drive other downstream biological processes. So in terms of a conveyor belt of candy, then we can think of ATP production as the way of generating the intracellular bonbons of ATP that are then consumed downstream by downstream processes. So when we look at this series of modules, we can see a couple takeaways right away. So one is that the cell works really hard to maintain ATP-ADP homeostasis. So this ratio is relatively fixed in most cells. And the processes that consume ATP are relatively insensitive to environmental changes. So another way of saying that is that this is pretty steady or steady state. This doesn't change very much. So I'm drawing an X to say that this changes relatively little in response to external fluctuations. Likewise, processes that consume ATP, things that the cell needs to do to run its biology, again, tend to change relatively little in response to external changes. The things that are available to change then that we have left are the pathways of ATP production and the way that ATP production is distributed between glycolytic ATP production and oxidative ATP production. So in most cells, again, or in all cells, this would apply, that in a cell that's running both pathways at the same time, the rate of ATP production in total can be summed as ATP from glycolysis and ATP from chemiosmosis and also substrate level phosphorylation. And then as we've discussed yesterday, in most cells, the rate of ATP production is exactly equal to or roughly equal to the rate of ATP consumption. So would anyone disagree with anything that I have just said? This all sounds pretty reasonable, right?
Wouldn't there be more production compared to consumption?
Wouldn't there be more production compared to consumption? Why? Simple?
I mean, you would want to have some reserve, right, in case of emergencies.
That's a really interesting point. Do you have a rainy day reserve of ATP in a cell, in a typical cell? If you were talking about your bank account, the answer would be absolutely yes. You would think about wanting to have some sort of backup or reserve of ATP in the cell. But in reality, the cell doesn't do that. It can't really do that. If you remember back to David's comment yesterday or this morning, the human body turns over about its own body weight in ATP per day. There really isn't a buildup or a spending of an ATP level. So that actually does stay pretty constant. And then what that means is that to maintain a steady state level of ATP, you have to have matching rates of production and consumption, which is what we see in most cells. So the only real buffer that you have to ATP phosphorylation then is like the creatine phosphate system or some other phosphorylating system that in a very short term can provide a little bit of capacitance to a sudden drop in ATP, for example, during sprinting, where, as we talked about yesterday, oxygen supply is too slow to allow OXPHOS to make up the ATP that's missing. So creatine phosphate then transfers its phosphate groups to ADP to regenerate the ATP. But in general, as a whole, no, there is no buffer of ATP in a cell. There's no rainy day fund. It uses the ATP as fast as it makes it to maintain steady state levels. But that's a really good point to make because it's counterintuitive to the way that we think about. So I often express ATP production and consumption in terms of budgets. And a budget and a bank account is the wrong way to think about cellular ATP production and consumption. Any other points, questions, disagreements? Okay, so I'm not going to run this next exercise as a breakout session. But let's go through a little thought experiment, and we can just do this kind of individually and then brainstorm as a group. So I'll show you some traces of an unknown inhibitor in an orange trace that's present from the start. So here's our control trace in black. I just drew these. These are made up traces. But the point that they illustrate, I hope, is clear. So we have an inhibitor that has the following effects on the respiratory trace. And if we look at the acidification trace, again, the black trace is the control. The orange trace is the presence of the unknown inhibitor. Let's take just a second to think about what could explain this. Feel free to talk amongst yourselves, use the chat window. We'll just keep it in the larger group for now, though. So let's take a minute or two to think about what sort of intervention could explain both of the effects on the respiratory trace and on the acidification trace. And feel free to brainstorm aloud or in the chat window.
Seems to be something that suppresses the ability to generate ATP through the extracellular acidification pathway or glycolysis.
Sounds good, Kevin. So what, in that vein, what type of thing would suppress the ability to make ATP through the same pathway that generates acidification?
Possibly a lactate dehydrogenase inhibitor.
Lactate dehydrogenase inhibitor. Is that something that everyone else agrees with? Inhibitor of glycolysis, PK?
Why does the OCR go up? So it's shifting respiration towards mitochondria.
Because the ATP synthesis from glycolysis is now not, yeah, it can't compensate. So the pyruvate goes into the, there may be more pyruvate if you inhibit lactate dehydrogenase.
It could be
So
on the
even
front end, like a glucose
dehydrogenase,
inhibitor.
see any
Sorry,
change?
Colin. I heard
Yeah, it could be on the front end, so it could be a glucose import inhibitor or even a hexokinase inhibitor.
So I guess what I did not say is what substrate these cells are running on, but let's assume that it's glucose.
So I was thinking, like, do you think that this scenario could be possible if we put, like, for example, these are the cells, cultured cells, and we increase the oxygen concentration in the incubator and forcing cells towards the oxidative phosphorylation rather than glycolysis?
So would increase, so these cells are all under air conditions, so we're not actually manipulating oxygen concentration exogenously to the experiment. So let's assume that O2 is saturating. And an increase in oxygen tension wouldn't explain the acidification result. So far we've got some sort of glycolysis inhibitor either at the front end, either in glucose transport or activation, or at the back end at lactate dehydrogenase. Any other possible explanations?
Some responses in the chat window.
I see. 2-deoxyglucose, yes. So thank you, Neil. So that again falls into the category of inhibition of glycolysis at the front end, where you're clogging up glucose 6-phosphate or 2-deoxyglucose 6-phosphate, and you're inhibiting downstream steps at phosphoglucoisomerase. Okay, so let's just back up a second and look at what we're observing. So it was correctly observed that you see increased respiration in the presence of the unknown inhibitor, but that otherwise this trace is exactly the same. Something strange is going on over here where we see, in response to oligomycin, we see what we've seen before, where as you decrease ATP-linked synthesis, respiration-linked synthesis through oligomycin addition, you see a corresponding increase in acidification, suggesting that you are shifting ATP production from oxphos to glycolysis, and then it stays high as you make additional port additions. The orange trace, you see a line that fluctuates with additions. And again, I drew this, it's not necessarily to scale. But if you have, as let's say, inhibited glycolysis, what explains these changes with the additions that are made here? How do we explain that trace? No further upregulation of glycolysis. So if we agree that we've inhibited glycolysis, changes in glycolysis cannot explain the changes that we see in the acidification trace here. So what's left?
Are you asking what's left to cause changes in the pH?
Exactly. So changes in pH are always the result of changes in proton concentration. How are we facilitating a change in proton concentration when we have blocked proton production through glycolysis, through the production of lactate from glucose?
Are you feeding these cells lactate?
These cells are only being fed glucose.
No, I mean, could the orange addition be feeding with lactate that would consume?
Could they be metabolizing the lactate that was generated at the beginning? Is that what you're asking, Daniel?
Yeah.
I suppose it's possible, but right here, that's not what's happening. I hesitated because there are a number of cell models where you can make them lactate catabolism competent, but you have to do that by growing them in lactate so that they start expressing transporters and things that help them catabolize the lactate. So a lot of cells are not lactate competent to begin with, but you can make them that way. But these cells are not lactate competent or lactate catabolism competent. And this is too short a time for that to have happened anyway. So what could explain changes in pH as a function of changing additions during a typical respiratory control experiment?
CO2 production?
CO2 production. So elaborate, Colin.
I don't know if I know the exact mechanism behind it, but I do know when I was learning Seahorse, they said if you ever saw an increase in respiration and pH, it could potentially be CO2 production.
Can anyone explain why?
You have CO2 production when you spin around the Krebs cycle?
Yeah, exactly. So you make carbon dioxide in the Krebs cycle. What happens to carbon dioxide in aqueous solution?
Complexes with water and then gives off a proton.
Yeah, it hydrates
Carbonic acid.
to carbonic acid, and then that carbonic acid dissociates almost immediately into bicarbonate and a proton. And so carbon dioxide is, depending on the system, either a small or a large contributor to total acidification that occurs as a result of a cell catabolizing substrates. So this is in fact lactate dehydrogenase. But one of the points that I really wanted to make here was that there is more than one contributor to proton production in a cell that's catabolizing substrate. So a major one, yes, is lactate production, but carbon dioxide also contributes. And you'll notice here in this made-up example that it contributes proportionally to the respiration rate. And so this is another way that you can tell that this is likely due to respiratory CO2, because the production of the protons that you see appearing in the ECAR trace here mirror or roughly match the respiration rate that you see in the respiratory trace. Does that make sense to everybody? Yes. So in aqueous solution, carbon dioxide readily dissociates after hydration to bicarbonate and a proton, which is a meaningful source of acidification and a meaningful contributor to the extracellular acidification signal that you'll see here as well. Okay, so this is actually an example that we just looked at, but let's revisit it. Does anyone remember what this inhibitor was, shown in blue here and here?
Protein synthesis inhibitor?
Protein synthesis inhibitor. Yeah, and Gulsen, am I pronouncing your name correctly?
Gulchen.
Gulchen. Can you remind us of why a cyclohexamide or a protein synthesis inhibitor would have these effects on the trace?
Because it's reducing the energy demand overall, and that's why the OCAR is going down. But then towards the end of the assay, that the effect that we also observe, especially on the ECAR, indicates that cells are not doing that great at the end of the assay.
I think that about sums it up. Yeah, so normally protein synthesis consumes ATP. If you block protein synthesis, you'll block that source of ATP consumption. You'll lower ATP demand. Because ATP demand is tied to ATP production, those two rates at steady state are equal. A decrease in ATP demand will lead to a decrease in ATP production. A decrease in ATP production leads to decreased rates of respiration and glycolysis. And then we talked earlier about the potential meaning of the fact that these traces are not equal. These traces are not equal. They might be meaningful differences. They might reflect artifacts of the continued presence of my. And could everyone make sure to mute their mics? Okay. So overall now, we have a sense that there are two pathways that are associated with ATP production. Those pathways are also associated with the indirect metabolites that we measure, namely the disappearance of oxygen and the appearance of protons in the medium. And so when we're talking about whole cells, we're always talking about the extracellular fluxes. That terminology gets a little bit inaccurate when we talk about isolated mitochondria, but it's still the disappearance of oxygen from the medium, the appearance of protons in that medium. So this is a diagram from the paper that we published in 2017, really just describing those pathways and then the sources of each of the things that are measured. So of course, the things that we care about are ATP made in glycolysis. We make a little bit of ATP in the TCA cycle at succinyl-CoA synthetase. We make the majority of the ATP in the cell most of the time through oxphos. We hate using the term oxphos. We do it all the time anyway. Please bear with us. Okay, so the beauty of the seahorse is that it allows us to associate the rates of ATP production that are actually relatively hard to measure directly with the rates of associated fluxes of protons, and oxygen. And those are relatively easy, again, to measure. So Martin showed you this trace yesterday. When he described it, he said, nothing much happens, nothing much happens, nothing much happens, something happens. But what I would like to point out to you now is that actually the nothing much happening here is meaningful. So you see a small but meaningful acidification rate under the same conditions that these are mitochondria respiring before the addition of rotenone here. And so like we just discussed before, what is the source of the acidification that's happening here? Remembering that these are isolated mitochondria, there is no glycolysis present. So let's connect the prior thought to this thought. What's the source of this acidification?
It's in the chat.
It's in the chat. CO2. Thank you, Mega. Yeah, so it's CO2. The CO2 that comes off of pyruvate dehydrogenase and a couple other enzymes within the citric acid cycle. So the fact that this has always been here is something that we sort of ignored for a number of courses, and then we realized that it was in fact meaningful. We could measure it, and then we could correct the rate of acidification to isolate glycolytic rate by subtracting off the respiratory carbon dioxide production rate. So the take home from this slide, I hope, is that you have rates of ATP production that are each associated with, in some way, measurement of an extracellular flux. And so the way we treat the extracellular flux then allows us to work backwards and estimate the rates of ATP production that underlie the measurements that we actually make. The inverse of that is that the respiration rates and the acidification rates themselves are imperfect representations of ATP production. And so that thought would tell you that looking at the rates themselves is often insufficient to understanding the energetic rates that underlie the measurements. So we have to treat these rates, correct them, and gear them appropriately in order to understand ATP production. And so once again, sorry, one second, let me close my office door. We have too many Zoom meetings going on at once. Okay, so this is again a simplified version of the simplified version that was on the last slide. Throughout the papers that we have published and the diagrams that we've generated, we always depict glycolysis in blue and things associated with oxidation in red. So when you see blue and red, we try to keep those consistent. So we have signals. And really what a seahorse machine does, or any other machine that can measure a simultaneous acidification signal and an O2 signal, is that we're measuring tensions, we're measuring levels. So the fluorescent probe in the seahorse plate is measuring oxygen tension as a function of time, and it's measuring pH as a function of time. So when we have the oxygen consumption rate, we have a stoichiometric rate right away. By converting the oxygen tension per time to a rate of oxygen consumption, we have molecules of oxygen and we can predict molecules of ATP, or mole to mole, however you want to think about it. But for the acidification signal, it's measuring pH. So this is a logarithmic signal. It's not immediately comparable to stoichiometric proton production. There's obviously a relationship between the pH of the medium and the concentration of protons in the medium. And to uncover that relationship, we have to convert the pH change over time to a proton production rate. And so one of the things that I know has tripped up participants in the course in the past is the way that Seahorse, or the way that the Agilent software labels this rate. And so you'll see it labeled as ECAR, which the units are pH change per minute per whatever microgram protein. But then they also have a proton production rate or a proton efflux rate. Those are essentially the same thing, but they're all measuring proton production. They're just doing it in different scales. So pH scale is logarithmic, whereas proton production rate is linear. And so in order to treat these measurements to understand ATP production, you first have to scale the pH measurement and convert it to proton production rate. And that's easy to do. You calibrate your assay medium against a known addition of HCl or whatever acid. And then you build a relationship of buffering between your assay medium and pH. And then so working backwards, you can convert your pH per minute change to a proton production rate. So once you do that, then you have two linear rates. Those linear rates are comparable, except that they're both contaminated by non-ATP associated sections. And so then you have to correct off those sections. We do that for the proton production rate by subtracting off the carbon dioxide contribution, which we understand by looking at the respiration trace, or the oxygen consumption trace. And then we correct the respiration rate in the way that we have, that David and Martin have alluded to earlier, where you can isolate the ATP-associated oxygen consumption by the before and after oligomycin. And you can isolate the non-mitochondrial oxygen consumption by the before and after rotenone and antimycin, or rotenone and mixithiazole. So we can understand of the total oxygen consumption rate, how much of that is phosphorylating, how much of that is proton leak, and how much of that is non-mitochondrial. And obviously, if we're concerned about how much ATP is being produced, we care about the phosphorylating portion of that rate. So once we've cleaned up our linear rates to isolate the ATP producing portion of each, then it's simply a matter of multiplying that rate by the gearing ratio of how many ATPs were produced per unit of chemical or metabolite measured. So for glycolysis, it's the ratio of ATP generated per lactate produced. And for oxygen consumption, it's the ATP produced per O consumed or per O2 consumed. So the P to O ratio classically uses just one oxygen molecule. After you weight these correctly, then you end up with comparable units of ATP production. And then finally, you can sum those rates to understand what the cell is doing as a whole. So one of the really interesting aspects of understanding ATP production is that you'll find very few examples in the literature where whole cell metabolism looks at both glycolytic ATP production and respiratory ATP production simultaneously. You'll see many, many examples in the literature of looking at one or the other, or looking at oxygen consumption and, for example, lactate production, and trying to have and make qualitative assessments or estimates of how metabolism might be changing under different conditions. But again, I think there are very few examples where you're actually comparing ATP production from the two different pathways in the same units. And so that's one of the real advantages of using an instrument like this, where from these two signals, we can fully understand within a set of model assumptions what a cell is doing and how much ATP a cell is making per unit time. Are there any questions so far? All right. So the mechanics of the conversion look like this. Bear with me. So we mentioned earlier that the rates that we can measure on a seahorse or similar instrument are essentially pH and O2, oxygen tension and pH. And so using the steps that we just talked about, we can convert the acidification signal into a proton production rate. We multiply that by the gearing of ATP produced per lactate generated. And to get this back all the way to the raw measurements on the machine, we can deconvolute our glycolytic proton production rate to this, which expresses the fact that you're measuring an extracellular acidification rate. To convert that to proton production from pH changes, you divide it by the calibrated buffering power of your medium. So this is the pH change that results from addition of a known amount of protons, or a change in the proton concentration of the medium. And then you subtract from that all of the acidification that's coming from carbon dioxide. And that's a function of this term, which is the mitochondrial respiration, which is the total minus the non-mitochondrial respiration. You multiply that by the ratio of protons you expect per O2 consumed for that substrate. So this is just a function of the stoichiometry of complete oxidation of that substrate. And then you have to add in the dissociation of carbon dioxide to bicarbonate and a proton. So under the conditions of most experiments, so pH 7.4 and 37 degrees, about 96 or 97% of the carbon dioxide will fully convert to bicarbonate and a proton. But the remaining 3% then needs to be accounted for. And so initially when we set up the system, we were always about 3% short. And then we were reminded by Andy Nielsen at Seahorse that we had not yet accounted for the fact that there's equilibrium between carbon dioxide, carbonic acid, and then bicarbonate plus a proton. So this incorporates the combined dissociation constant of those two reactions under the conditions that most experiments are run under. So yes, there are a lot of terms here, but it actually breaks down relatively straightforwardly that you are taking the total acidification and you're subtracting from it respiratory acidification to isolate glycolytic proton production rate. And then when we look at the rest of this, the same idea applies, that you're taking each portion of the respiratory trace, so for example, mitochondrial part, and the part that's coupled, the part that's phosphorylating, and you're multiplying each by the portion of the P to O that's responsible for ATP production during that part of the combined pathways of ATP production. And so the most prominent one for respiratory or oxidative ATP production is, of course, ATP synthesis at the ATP synthase. So that requires the coupled respiratory rate times the P to O for oxphos. We also take into account the ATP production rate from glucose catabolism that yields pyruvate that is not actually converted to lactate. And then finally, we also take into account the ATP that's made at succinyl-CoA synthetase as a function of the total respiratory rate. So let me back up 1 second and divide this into the two portions that we've assigned it. So we have one section here that has two parts. We have a second section here that has two parts. So we consider both of these parts to be J-ATP, this will take me a long time to write, glyc. We consider all of this to be glycolytic ATP production, which is a bit of a departure from how it's often expressed in the literature. But I would argue that it makes more sense because remember that when you're catabolizing glucose, as this glucose runs through glycolysis to pyruvate, you're making ATP independently of whether that pyruvate goes on to be oxidized in the TCA cycle or whether it goes on to be reduced to lactate. And so a typical definition of glycolytic ATP is based on lactate production, which is true, is the majority of glycolytic rate. But there's also a small portion of that rate that's never seen as lactate because it's oxidized in the TCA cycle. And so if you would agree that all ATP that's made in glycolysis should be defined as glycolytic ATP, you would also agree that not only do you have to consider the pyruvate that goes to lactate, but you also need to consider the pyruvate that then goes on to be oxidized. And that is why we have both of these terms here. So this is the pyruvate that's gone on to lactate. This is accounting for the pyruvate that then gets oxidized. And then these two together are glycolytic ATP production. Still here?
Yeah, I have a question.
Go ahead.
If then you have terms for coupled OCR, doesn't that already count the pyruvate that gets oxidized in the TCA cycle?
No. So what we're accounting for here is actually the ATP that's made at substrate level glycolysis. So the coupled ATP production at the ATP synthase is after electron so after reducing equivalents enter the mitochondria from glycolysis, we count that as oxidative ATP, and we count pyruvate oxidation as oxidative ATP. But this ATP still needs to be accounted for, and that's not accounted for by this P to O. Okay. And then just to finish that thought, so we have the two parts of glycolytic ATP production. And then we have the two parts of oxidative ATP production, one of them chemiosmotic at the ATP synthase, and one of them substrate level at succinyl-CoA synthetase. And so these two P to O ratios use different portions of the respiration rate because for ATP synthase operation, we care about just the coupled part of the rate. We don't care about oxygen consumption that goes through proton leak. But for substrate level phosphorylation at succinyl-CoA synthetase, we do care about that portion because that's the portion that's seen by the TCA cycle. That portion drives substrate past succinyl-CoA synthetase to phosphorylate ADP to make ATP. And so in order to run the calculation, you need to use the whole trace of a typical mitochondrial respiratory control experiment. So when people ask questions like, well, what's the ATP production in my cell? The answer is always, it depends on the condition. But you can actually calculate the ATP production rates at any portion of this trace as long as you have the rates after oligomycin addition to define coupled respiration, and then the rates after mitochondrial poisoning to subtract off non-mitochondrial respiration. And so the whole trace here gets utilized. So the trace that I'm showing here is a little imperfect for this example. We've added a blank because we've added different compounds in other experiments, but we have all the elements that we need. So we'll typically run, or at least for these experiments we collected a basal rate before we added exogenous substrate. We added glucose. So this is the acidification rate. We added nothing here, just DMSO. And then we collected oligomycin and rotenone and mixothiazole rates. And so for the acidification trace, we have the same data that we have for the respiratory trace for each of the different signals. So using one trace's worth of data from both of these signals, we can run this conversion to generate total rates of ATP production during different portions of our typical respiratory control experiment. So when we look at the raw data, when we look at these raw traces of extracellular acidification and oxygen consumption, you can see qualitative changes happening, right? As we have talked about prior to now, you can see when you add glucose, maybe we haven't talked about this yet, but when you add glucose, you see a drop in respiration that corresponds with an increase in acidification. What's happening there? What do you think's going on with that change? Cells have no glucose in the beginning, and then we add glucose, and we see this flip-flop in the rates. What's a likely explanation?
Traditional glycolysis.
What is traditional glycolysis?
Glucose to pyruvate through LDH.
Sure. Yeah, so why are these rates changing in the way that they are, Colin?
When you have ATP production from glucose, the need for respiratory generated ATP goes down.
Yeah, I could not have said it better myself. Yeah, so when you offer the cells a way to run glycolysis by introducing a substrate that glycolysis can use, like glucose, right here, the cells are making all their ATP or most of their ATP through oxphos. When you offer them glycolysis as another means of making ATP, it will do so. And the need to drive respiratory ATP production drops. And therefore, OCR drops a little bit. And you see, in addition, an uptick in extracellular acidification, indicating an increase in glycolytic rate as well.
What's the substrate before adding glucose?
There is no exogenous substrate. So the cells are clearly catabolizing something. So we have speculated that it's either intracellular glycogen or other intracellular substrates. So even though we starve the cells prior to addition to the seahorse to try to minimize the contribution of endogenous substrates, they're clearly able to poke along using those for a while. So the answer to your question is that we don't know, but within the fit of the model, the substrate is consistent with either being glycogen or with being the sum of other endogenous components whose H plus to O ratio satisfies the assumptions that we've made for the calculation. But it would be interesting to try to figure out what these substrates are, and it is probably cell type dependent, and it's probably very sensitive to the treatment that the cells get prior to being assayed.
I have a quick question.
Go ahead, Alex.
When you're doing all of these calculations for ATP, is there anywhere where you account for the ATP requirement for glycolysis and that consumption of 2-ATP? Does that ever get added in?
Absolutely. So that is incorporated into the P to lactate.
Gotcha. Thank you.
So but that also applies to respiratory ATP production. So there is energy consumption in the form of proton loss as you transport phosphate and ADP into the mitochondrial matrix, for example. And so all of those gains and losses, the net effect yields the various gearing ratios of ATP per either lactate produced or O2 consumed or O consumed. So the net P to O ratios reflect, all of the ATP production and ATP consumption that follows from catabolism of these substrates. But that's a good question. Okay, so when we look at the raw rates of a typical respiratory control assay, we've just talked about how all the rates that you need to define the various portions of respiration and acidification are contained within the typical respiratory control ratio assay. This is often called the mito stress assay in Agilent nomenclature. But before Agilent packaged it this way, Martin and David conceived of it as the respiratory control ratio experiment that we've discussed earlier. So you need a basal rate, and that basal rate can be defined by your experimental design. So that's with or without substrate, but typically before any sort of drug intervention or any of the making the mitochondria jump through the hoops that we do with oligomycin and with rotenone and mixithiazole or antimycin. You need to define ATP phosphorylating oxygen consumption. You do that by adding oligomycin. If you want to understand the maximum respiratory rate, you uncouple with, for example, FCCP. And you need to define non-mitochondrial respiration by inhibiting electron transport with a combination of mitochondrial poisons. And then what these plots on the bottom show you is how we convert those rates to summed ATP production as a function of the whole cell. And one of the interesting things to note is that when we do that, you can watch the ATP production changing as a function of a total rate in the cell. So for example, under the basal conditions we've defined here, there's no exogenous substrate. And you can see in this left trace, the red portion is oxidative ATP production. The blue part is glycolytic ATP production. So whatever the cells are surviving on, it is almost entirely respiratory. As soon as we add glucose, we provide a means for glycolytic ATP production. So glucose or glycolytic ATP production shoots way up, and respiratory ATP production declines a little bit as a result. But you'll notice that the net ATP production rate is actually a little bit higher than it was before we added glucose. When we add oligomycin to inhibit the ATP synthase, these cells, these are C2, C12s, have sufficient glycolytic capacity that they can basically account for all the ATP demand that was taking place here and meet it with glycolytic ATP production here. So what you're watching as a course of this experiment, or a course of this trace, let me get rid of all of my things and go back to here, is that you're effectively watching ATP production shift from respiratory or oxidative ATP production towards glycolysis as you add glucose, and then completely by glycolysis as you inhibit the ATP synthase. And because we can define on a more sophisticated level the P to O for each of the phosphorylating reactions through substrate level phosphorylation and through chemiosmosis, we can actually define the portions of ATP production from pyruvate that gets oxidized, from lactate production, from the succinyl-CoA synthetase in the dark red up here, and from chemiosmosis here. So you can actually see proportionally where ATP is being made and at what rates. Okay, so
I will have one, sorry, Donna, I have one question on the figure C. I mean, from the beginning, you said that, you know, the ATP consumption is not really changing. So here, like, you know, on the C, when you add the glucose, so the cells are like producing like more ATP, obviously because of the glucose addition. But so because there was a discussion that, you know, the cells do not generate ATP if it doesn't need it. So in that case, so the basal is the part that cells need. So apparently those cells like need more energy, or they just like produce just glucose over there.
What I said earlier was a little bit of a lie, and that, I mean, so if we convert that statement to a statement of control, what I said earlier would be consistent with the idea that control over the flux is completely by, or primarily by ATP demand. But what this would tell you, the increase that you see here, would suggest that a small portion of the control over flux is actually on the ATP supply side. So this would demonstrate that it's not entirely true that ATP demand controls the rate of, controls the flux. That you can push more ATP supply, and that also has a slight effect towards increasing the flux. As well.
That was my question, actually.
That was your question?
Yeah, that's what I was asking. Okay.
Okay, yeah. Perfect. Yeah. Okay, so one of the really important things to take away whenever you're looking at seahorse data, whenever you're looking at published literature, whenever you're looking at your own data, is that even though these rates both reflect ATP production, they are contaminated and they're not comparable to each other. So we'll use the apples and oranges analogy a lot. So if you try to compare OCAR to ECAR, it's like trying to compare apples to oranges. And if you want to make sense of these two rates together to understand total cellular ATP production, you have to convert them into the same unit. Then you're comparing apples to apples. And so to illustrate that, I've just mocked up these. So these are actual data. But let's look at strengths and weaknesses of these data sets. So if you're just looking at the acidification trace and the respiratory trace as it would come off the seahorse, yeah, you can plot them together. Yeah, you can plot them against one another. This OCAR to ECAR plot is actually one of the default outputs of the current software. But this relationship is arbitrary, right? It is set entirely by what the buffering of your medium is. So if you have a very strongly buffered medium, you'll have very small pH deflections and therefore relatively low acidification rates. But your respiration rate, of course, is not affected by the buffering of your medium and will be the same independently of that, or largely independently of that. So the relationship between these two rates is arbitrary and partly determined by your experimental conditions. Your acidification rate is confounded by carbon dioxide, as we discussed earlier. There's a small contribution of pyruvate that contributes to glycolytic ATP production that is not measured as lactate. So you miss that if you're only looking at the acidification trace. Another difficulty in comparing these two rates relative to understanding ATP production is that they're geared differently. So the stoichiometry between O2 and ATP and H plus and ATP is different. And then for the respiratory trace, we have proton leak that confounds our ability to understand respiration-linked ATP synthesis unless we do something like add oligomycin. And so it's easy to see from these objections that it's really hard to get an intuitive sense of what a cell is doing just by looking at the raw rates. So what you can actually do is get a sense of how the rates are changing with different additions. And so there are comparisons that can be made, but those comparisons have to be qualitative or at best semi-quantitative. Whereas when you make the conversions to ATP production rates, then you open up a whole new level of the ability to analyze and understand the data that come off the seahorse. So we haven't actually changed anything about the measurement. We've just treated the measurements differently. And by defining the ATP producing parts of those and gearing them appropriately, you gain an incredible amount of information from that conversion. So one, you can directly compare the same units to one another. By extracting the ATP producing portions of each of your measurements, you remove most of the confounders that we objected to up here. You account for the cross contribution of the signals to one another. So we talked about carbon dioxide contributing to ECAR. We talked about pyruvate production contributing both to glycolytic ATP and to oxidative ATP. Once you do the correct gearing, you can directly compare glycolytic and oxidative ATP production because now these units are the same. And then finally, if the goal is to understand what a cell is doing and how a cell is responding in terms of its total ATP production rate to an intervention, to a mutation, to a pharmacological change, you can sum these rates to understand total cellular ATP production. So two different ways of plotting are kind of your typical column plot or your 2D plot. And we use both of these in the way that we analyze. We already have the ability in a typical respiratory control experiment to elicit maximal respiration, as we've talked about, where you add, if you're adding your substrate in flight, you do that, you add oligomycin, you add FCCP to elicit maximal respiration, and then you define that by adding mitochondrial poisons. We've developed a similar structure for analyzing maximum glycolytic capacity, which your respiratory control experiment won't elicit for reasons that I do not have time for, but I'm happy to get into it during the practical, where we change the experiment a little bit. So we'll add our substrate, We omit oligomycin because you'll actually allow the ATP synthase to run backwards, therefore acting as an additional source of ATP demand to try to elicit the maximum glycolytic rate possible. We poison the mitochondria. And then we add monensin, which is a sodium channel cycler at the plasma membrane. So we're trying to invoke a futile ATP consumption rate in the ability of the plasma membrane to reestablish a sodium gradient. And you can see in the acidification trace here that it actually elicits a slightly higher rate than that with rotenone alone. And so we're using this system then to define the maximum acidification rate and convert that to the maximum glycolytic rate that the cell can support. I swore I wouldn't go over time. I'm going to go over 1 minute because this is a really important point to make. Because of the historical way that bioenergetics has been approached, typically either as a respiratory issue or as a glycolytic issue, measuring glucose uptake or lactate production, we've tended to think of the two pathways differently. But that's a little bit like thinking about your geographic position in terms of either latitude or longitude, but not both. So if we look at, for example, a typical respiratory trace on the y-axis, we have our rates before we add a substrate, we add glucose, respiration drops a little bit, we add oligomycin and FCCP, we elicit a maximal rate, and we kill everything with a mitochondrial poison. So this is the information that the respiratory trace converted to an oxidative ATP production trace gives us. If we add in glycolytic ATP production, we suddenly see a much different picture of what the cell is doing relative to its ATP production rate. And we can define certain parts of this. So the maximum respiration rate and the maximum glycolytic rate, we can define as the boundaries of this theoretical space. And again, we've determined experimentally that these are the maximum rates that each of these pathways can support. Keep in mind though that the glycolytic maximum is a true ATP synthesizing maximum, whereas maximal uncoupling is really just maximal respiration rate. Being uncoupled, this is not ATP producing. So this is a theoretical production of ATP that's possible through respiration, but not empirically demonstratable. Where those two intersect is a theoretical then capacity of what the cell can do if it were to run both respiration and glycolysis at their respective maxima. We don't know that a cell can do this, but we can define this theoretical space nonetheless. And we call it bioenergetic space. And then we can do really interesting things with that space. For example, this red dot is a cell respiring under conditions of the presence of glucose. But what this diagonal line tells us is the same ATP production rate that can be supported by a changing ratio of oxidative ATP production to glycolytic ATP production. So for example, you could generate the same ATP production rate through any one of these combinations of glycolytic and oxidative phosphorylation, as long as you don't exceed the maximum in one direction or the other. And this tells you essentially how flexibly the cell can use glycolytic and oxidative ATP production to meet a particular ATP demand or to match a particular flux of ATP turnover. I'll skip these sections for now. So I hope that you agree that by now, or after you think about this a little bit, that you can identify the major mechanisms of extracellular proton production and O2 consumption in cells catabolizing glucose, that you can define glycolytic and oxidative ATP production, that you're able to calculate total cellular ATP production given oxygen and proton fluxes under rate-defining conditions such as a respiratory control trace, and that you can use those rates to determine bioenergetic indices and capacity. Any questions?
Yeah, we had some discussion on the idea that adding in oligomycin and then FCCP sequentially can change the sort of bioenergetic state of the mitochondrion.
Yes.
I was wondering to what extent that's affecting this type of assay where you need to do both to get at all of the terms that you use.
That's a really good question. And so let's see what I can show you. So these don't actually show you that because we didn't care about eliciting maximal respiration here. But as David alluded to earlier, based on my comment, that the way to test whether uncoupling after oligomycin can actually elicit maximal respiration is to compare that to when you add those two things simultaneously. So for example, in C2C12's running glucose, I've seen in the lab that C2C12s just given glucose can drive maximal respiration to about the same level as basal if oligomycin is added first and then FCCP is added second. But if you add them together, you actually do get a little bit of spare respiratory capacity that isn't detectable if you add the oligomycin and FCCP sequentially. So that comparison is a good standard pilot experiment to run when you have a new cell type or you're assaying cells under a new condition, if you care about maximum respiration. And so if you care about the ability of the cells to drive the maximum respiration they can under the conditions you're assessing, that's a good comparison to have as a default sort of setting up experiment.
I have another question relating to the stoichiometry between oxygen consumption and proton translocation.
Sure.
Since it's going to be different for succinate and NADH, for example.
Yes.
How do you combine those two terms in your calculation, since a full turn of the TCA cycle is going to add to both of these?
So the P to O ratio of NAD-linked substrates and succinate will be different. And that's accounted for in the assumptions that we make for the calculation. So I'm happy to go through those offline. What was the second part of your question?
Well, how do you combine the NADH contribution and the succinate contribution to your HO ratio?
Okay, so the H plus to O ratio is going to be simply a function of the stoichiometry of the complete oxidation of the substrate. So it doesn't matter what pathway it goes through.
Well, succinate pumps like what, 6 and...
Sorry, I think we're talking about different things. Yeah, so if you're talking about proton pumping, absolutely then yes. So when we published this paper, we generated a table of where all of the protons were coming and going and how many protons were pumped per NAD-linked substrate, which is 10, or succinate-linked substrates, which is 6. So that ends up in the final P to O ratio for those substrates. And so for every turn of the TCA cycle, you'll have some electrons entering at succinate dehydrogenase and some electrons entering at NADH. And those are all accounted for per mole of substrate. Okay.
And I guess the question then is, in a given turn, is that, are we assuming that each time we add glucose to something that
Sorry, Daniel, I just lost you.
glucose is... Sorry.
I just lost your entire question.
I guess in a full turn, you oxidize NADH, or in a full turn, you generate NADH and you generate and then oxidize succinate. And that's part of your calculation, but
yes.
Are we assuming that this full turn has no anaplerosis, either coming in or going out, that's going to change that stoichiometry between NADH and succinate?
You raise a good point. And so one of the assumptions that we make, absolutely yes, is complete oxidation of the substrate. And so to the extent that's wrong, we have tried to work out how much the P to O ratio might be thrown off by incorrect assumptions. And as far as we can tell, it turns out that it's not much, especially within the error of the measurement of the seahorse. And so, for example, for glucose, like our OxPhos P to O is something like 2.486, and it goes down slightly if we assume some anaplerosis, but not very much. So it's less than the 10% error that you make on the measurement in the first place. And so to the extent that it's sort of in the wash of the error, we ignore it. But it's actually a really important point, because it means that if we have to make assumptions about what the P to O ratio is and what the H plus to O ratio is, there's the potential to miss biology that's happening in a way that is unexpected or that doesn't match our assumptions. So I would say that our model currently is well validated for specific cell types metabolizing glucose under conditions where we think they're not growing and where anaplerosis isn't running very quickly. So to the extent that actual biology will violate those assumptions, this model will match less well. So I would say that we're sort of at the level of, in physics, where you imagine that this horse is a sphere, right? It's obviously not a sphere, but it's fine if you're trying to model a herd of horses running across a plain. That's the level of resolution that I think we have currently, where it's actually not that resolved, but it's still a good way to estimate ATP production for cells in the seahorse. And there's still so much low-hanging fruit in that level of resolution that there's a lot of worthwhile experiments to do, while at the same time we work really hard to resolve our ability to not assume those P to O ratios or have better assumptions that apply more broadly than for the systems that we have currently designed.
No, it's really good to hear that that number, or H plus to O or P to O, I'm not even sure anymore, that ratio isn't very much affected by that.
As far as we can tell, it's not, but yes. So it will always be true that there's probably unexpected biology that these assumptions will miss. And that's then the really interesting thing to tackle next. Other questions before we take a break? All right, so why don't we take a break? I hope that you have access to the P3 Excel sheet. I will not be releasing the slides that I'll show until after the seminar, because it gives away too much of the narrative process. So don't be worried about that, but you should make sure you have access to the Excel sheet, because I will be asking you guys to manipulate that and then discuss effects of that manipulation in small groups, which we will do when we reconvene at 3:30. Going to go here. Okay, so welcome back. Does everyone have access to the Excel spreadsheet that was in the student folder? This is something
I have access to, what's it called? Could you remind me?
It's called something like 2020 P3 Excel sheet. Or
it's under programs.
Yeah, it's in the program.
It's in the programs folder. So does anyone else not have that Excel sheet? It's possible to follow along without it, but it's more fun if you can manipulate it in front of you.
I wonder if we can share that file in the chat window. Trying.
Maybe Ricky, could you look into that?
Yeah, do you want me to just share my screen or like?
No, you can actually share a file in the chat window.
Okay.
Yeah, thanks, Neil. All right, so we just kind of went over the principles of the ATP calculation, the portions of the calculation. So what we're going to do now is go through an Excel spreadsheet where I've put a version of the calculation that we use in a way that I hope is easy to manipulate and easy to see where all the parts add up and how we go from the raw measurements of pH and O2 to rates of ATP production that we sum to rates of total cellular ATP production. So if you were to open this file, it's actually 2020 P3J ATP. You'll see a file with a bunch of tabs. And the first tab is named Sample 1 OCAR-ECAR, and it'll look something like this. So I'll give everyone a minute to try to get there. Is everyone able to access this file and is open to this tab?
Yes.
Okay. Sounds good. I'm just looking for nods. Either you're fine or you're not willing to say that you're not fine, and I'll take either one. Okay, so what I would like you to do is this. You see in this top box, there's this section of original values. And what I would like you to do is not do anything before I ask. So all of these boxes and stuff are removable, but please don't remove them just yet. This is a play and we're going to go through it act by act. And so what I would like you to do is to copy these values from the protein cell to the ECAR total cell, and then paste them into this window. So control copy, control paste. And what you should see is that this column plot populates. And it should look like this. So what I'd like you to do next in our first breakout session, I'll release these questions. Go ahead and take a screenshot so that you'll have a copy of the questions to discuss in your breakout room. Please don't feel restricted to these questions, but the things I want you to think about when you're looking at these data include, how do these values compare to one another? What does this comparison say about rates of respiration and rates of glycolysis? And in your sort of intuitive assessment, or even if you think you have a quantitative assessment, which is this cell more glycolytic, more oxidative, or neither with respect to ATP production, and why? So let's take a few minutes to go into our first breakout room. Ricky, could you give us 5 minutes? So what can we understand from these data? What do we see here? What's happening here?
You can't compare those two bars because they're apples to oranges.
So we have what look like apparently equal rates of OCAR and ECAR, to use Agilent nomenclature. They look equal, but as Zimpel pointed out, the units are very different. So do we know that these are equal?
No.
No, maybe they are. They're probably not. But the visual appeal of things looking equal is an easy way to be misled. So what else do we know is going on? These are just raw rates. So this is the total rate of oxygen consumption, the total rate of extracellular acidification. So what do we know is underneath these values? What does the total oxygen consumption rate tell us? Or what is this a combination of? What do we have going on in, sorry, the red trace, the red column?
ATP synthase and
we have ATP
leak.
synthesis happening. We have proton leak. Anything else?
Non-mitochondrial respiration.
We have non-mitochondrial respiration. So we have lots of different kinds of oxygen consumption that comprise the red column. What about the blue column, the extracellular acidification?
Same thing.
CO2.
We have CO2, and we have, what else?
The pyruvate related, no.
lactate.
We have
slash
lactate production.
Okay, yeah.
There might be other sources as well, probably small, but possibly there. Yeah, so we have multiple contributors to each of these measurements. While these measurements look equal, their units are different. It's impossible to really compare them. Is there anything else that we can take away from these values?
I guess in a qualitative sense, the fact that we're assuming the mitochondria are 50% coupled probably means that there's a lot of glycolytic activity to support their ATP production.
Right, so we can infer that glycolysis is running. We can infer that respiration is running. We can infer from those together that both pathways are probably making ATP. Daniel, you're jumping a little bit ahead of me. That's fine. Yeah, so I've made some assumptions here that will become important later. So we have, at the end of the day, an implicit sense that both pathways are probably running, but we don't have a good sense of the proportionality of those pathways relative to ATP production. So I hope you agree with that. Does anybody disagree? Do we know more than that? Nah, I don't think we do. Okay. So we did these questions. So delete go ahead and delete that peachy box over the middle plot in your Excel sheet. You should see something like this. Is this the right way to think about these rates? Can we even do this? Can we add them? Is adding them useful in any way?
No.
Nah, this is garbage. This is terrible. What do these rates mean when you sum them together? They're not the same unit of rate. So it makes no sense. So we know that from this plot that we have some intuitive sense that both pathways are running, but the way to understand them together is clearly not to add them. Let's go ahead and delete that last box over the rightmost plot. You should see something like this. Is this the right way to think about it?
Only if you're comparing different conditions and you just want to see those conditions effect on one or the other.
Only if you're comparing different conditions. So what if this dot represents our control condition and this dot represents our experimental condition? Is there something you can say about that?
Yeah, that this cell has become more glycolytic and oxidative.
Yeah, so that ATP, so that something's gone up. Both rates have probably gone up. We see an increase in respiration relative to our control. We see an increase in glycolysis relative to control. Anything else? Go ahead, Daniel.
I was just going to say it's probably important to note the lack of deconvolution in the ECAR from CO2 that is produced in OCR.
It's crucially important to note the lack of deconvolution. So CO2 is likely contributing here. What else is confounding our understanding of how these two things are changing relative to one another? It looks at first glance like respiration has increased a little bit and glycolysis has increased a lot. Is that true? It could be true, but it could equally not be true. What's confounding this comparison?
Acidification occurs in the process of oxidative oxygen consumption.
Yeah, so you're right. Acidification will occur as a function of respiration that we can account for with carbon dioxide, which we haven't done here. So that's a contributor. There's a more sort of technical contribution here that's confounding how we think about these two rates together. That has to do with our units.
Well, the units just are straight up. They're not comparable whatsoever, right?
Yeah. Why are they not comparable whatsoever?
Log scale?
Because they're different units.
Yeah, one's a log scale, one's a linear scale, but more importantly, the pH scale will be, or the change in pH is completely dependent on the buffering power of your medium. If you have a strongly buffered medium, you might have the same change in glycolytic rate, but produce a very small change in acidification. Conversely, if you had an assay medium that had no buffering, you might have a change in acidification that's off the scale. Right, so the change in acidification rate is linearly dependent on the buffering power of the assay medium that you're measuring, that you're making your measurements in. And so the reason that I put this plot in the way that I have here is that this is one of the default output data pieces in the current WAVE software on the Seahorse machine. And one of the ways in which this is often labeled is as something like quiescent, energetic, glycolytic, and oxidative. And it's divided into quadrants. And those quadrants seem to be just the middle of whatever scale is chosen based on whatever axes fit the data set. And so the two things to point out are, one, everything we just said about how difficult it is to assess anything in particular about a point. You can make some qualitative comparisons, like when you have a second experimental sample, as long as they're comparable. But again, we explored how difficult it is to really understand how these two things are changing relative to one another when the scale itself is different and when the influence of your experimental conditions is different across these two measurements. So at the end of the day, it's really hard to interpret anything in particular from simply a plot of oxygen consumption and acidification. In the same way that it's really difficult to do anything, even though we know we're assessing some aspect of both glycolytic and oxidative ATP production, we just can't use these values usefully to try to combine those to get a sense of total cellular ATP production. So I hope if you take nothing else away from the afternoon, it's that. It's that to scrutinize your data and really try to understand what it can tell you and what it cannot tell you. Okay. So with that, why don't we go ahead and flip to the second tab, sample 1 JATP. So we see a similar setup. We see the exact same numbers, I hope, that we saw on the last tab. And what I'd like to do is the same set of operations. So let's copy all of the information from protein to ECAR from the original value section and go ahead and paste it in the sample section over here.
Are these Excel sheets linked? Because I see that it's already been pasted there.
Yeah, mine too. It's already done. It's already been done.
So I probably saved it that way. I've done a step for you already.
Okay, great.
I hope the boxers are still over the second plots, but if not, just ignore those for now. So what you should see is something like this. So in our second breakout room, which I'll ask Ricky to send us to in a second, go ahead and take a screenshot of these questions. Consider these and other questions that you might have as you interpret what you think is going on here. So I'd like you to try to figure out if the cell is more glycolytic, more oxidative, or neither with respect to ATP production. What's the percent distribution of glycolytic and oxidative ATP production? How would different oxygen consumption rates after oligomycin or rates after rotenonimixathiazole values change ATP production as you see it in this plot? Ricky, could you send us into breakout room? Do we have everybody? I think we do. So you guys were correctly loath to make an opinion about whether cells were oxidative or glycolytic based on the raw rates that we looked at in the OCAR-ECAR tab. Is your opinion clarified looking at this tab? Can you have an opinion about whether the cells are glycolytic or oxidative primarily based on these data?
Yes.
Yeah. Looks like, yeah, glycolytic.
Yeah. Looks like glycolytic. How much glycolytic? What magnitude of glycolytic is it?
Threefold more.
Yeah, so if we express this as a percentage.
77.
It's like almost 80% is contributed by glycolysis.
Yeah.
Looks like.
So we can say that this cell is roughly 80% glycolytic with respect to its ATP production. And then inversely, it's about 20% oxidative with respect to its ATP production under these particular conditions. Let's look at this last question for a second. So the rate that you input of 43 picomoles of O2 per minute per well obviously is not what we care about relative to ATP production, because we have the contributions of proton leak and we have the contributions of non-mitochondrial respiration, right? So this value that we get of about 80-20 glycolytic to oxidative is dependent not only on the rates themselves, but how much of those rates are phosphorylating. And so this last question was really addressed at letting you understand how much the correcting values, or the values that help you distinguish different parts of the respiratory trace, influence the oxidative ATP production that results. So how do different respiration rates with oligomycin and respiration rates with rotenone change oxidative ATP production, or change both of these? If you messed around with these numbers by plugging in different ones, what did you see as the result?
Increasing oligomycin puts a higher value on the proton leak. So we're seeing lower generation of ATP from the oxygen consumption, and vice versa for reducing it.
Yeah.
And then it was interesting for the rotenone, when we're inhibiting complex 3, decreasing that actually provides an increase to, increasing that value results in an increase in glycolytic. Is that sort of going back to glycolysis? How is that tied back to background non-mitochondrial oxidation, I guess?
Sorry, say that again, Carl. I think I followed you, but I want to make sure.
So that value is sort of the base non-oxidative or non-mitochondrial oxygen consumption. But increasing that value results in an increase in the glycolytic contribution. So where I understand that that value shouldn't contribute towards ATP production in the mitochondria, but why does it result in an increase in non-mitochondrial or glycolytic?
That's an interesting question. Why would increasing the non-mitochondrial respiration value do anything in particular to the glycolytic ATP production rate? The answer to that question is in how we calculate and then subtract off the portion of the acidification trace is attributable to carbon